a geometrical property, where, for example, if you have a system and you have the mirror image, and you try to overlap these two, you cannot, okay? Lord Kelvin just defined that this is a figure or point of group, and this is just a geometrical property, okay? However, when you have this kind of system, like these quartz crystals, and you have a linear polarized light, okay? And this polarized light goes through this quartz crystal. At the end, the crystal rotates the polarization, the plane polarization of light. Okay? This was observed a long time ago, more than the 100 years of the chemistry school, in 1811 by Arago, a French scientist. In the biography says he was a mathematician, physicist, chemist, everything. He was a very complete scientist. And after that, for example, Biot, one year after they worked together, they discovered a second kind of crystal that rotates also the linear polarization, the plane polarization of light, but in the contrary direction. It was a mystery for a long time until Pasteur arrived. Pasteur was more or less born at that time, as Arago and Biot discovered this phenomenon. And Pasteur solved the mystery. He was working with tartaric acid, and well, he found that it could exist in two different isomers, but with identical composition, okay? And he called, at that time, optical isomers, because one rotates the polarization in one direction and the other one in the other direction. He also found that the natural tartaric acid that is produced or found, for example, this is found in grapes, okay, rotates the light polarization only in one direction and always in that direction, okay? So he found that for this natural product from the nature has some kind of symmetry, okay? However, if you produce in the lab the same product, you can find that there is not any rotation. And again, it's because you overlap the rotation to the left and to the right, for example, of the linear polarized light. And this is because you have the two identical chemical composition system, the two crystals, and at the end, the sum of both, you recover the no rotation of it. Okay? So this, which is named optical activity, is a physical phenomenon that is associated just with this linear rotation of light when it propagates through these chiral systems. Okay? At that time, Pasteur called asymmetric systems. Well, at the end, well, it named this tartaric acid as the racemic acid, and from then is the name of racemic compounds, when you have both enantiomers in equal amount. Okay. Well, there are some kind of, one is, as I said, well, the racemic mixtures are obtained when these are produced in a lab, and always you will find the 50% left and the 50% right. Okay? Why? Because they have exactly the same composition, chemical composition, so there is not any difference of energy that can produce one over the other. So you always will find the 50 and 50. When the system, when these compounds interact with other systems, if these are not chiral, okay, the enantiomers have the same identical chemical reaction, chemical properties, and physical properties, except for the rotation of light. Okay, it's the only thing. Probably I should be in this place. Okay, and so these are called left, minor, 16th, the Levogero, etc. And the right. There are so many nomenclatures that I'm not exactly know why chemists use so many nomenclatures, but okay. So one important thing that I said was this like chirality is in the living world. And we are made by chiral parts, okay, building parts, like we just have less amino acids, we just only produce right sugars and so on, no? And all products, natural products, for example, if you want to see if the cocaine is pure, you can see that also the cocaine is chiral, you know, for example, in a right? Okay. This is a way to do that. Well, so another phenomenon associated with this optical activity and was found by Heidegger and Cotton, okay, and well, first Heidegger and then Cotton, they realized that, for example, this linear polarization light that goes through this chiral system at the end finish with an elliptical polarization of light, okay? And this is why, because, for example, if we consider the circular polarization of light, for example, this one, the light is propagating along this white line, and the electric field has two components that at the end finish with this rotation, okay, to the right, and also you can do the same with the left, okay? And we write like this electric field, this polarization. When you do the sum of both, okay, it's easy from here to see that you finish with this linear polarization again, okay? So at the end what happens is that if you have this linear polarization, which is the sum of the left and right, okay, one is going, the green one is going to the right and the other one is going to the left, and you put a chiral system, okay, what happens is that at the end this chiral system that absorbs energy, that absorbs light, okay, absorb preferentially in one direction, for example, the green one in this example, instead of the red one. At the end you finish with a difference in the amplitude of the polarization of light, and then you finish with this elliptical one. So more or less this is the way to do it. Now let me just talk a little bit about the chiral interaction, the chiral objects like our hands and our feet. Are called enantiomers. And for example, you have, as I said, if you have an ambient which is not chiral, which is a chiral, it doesn't matter which stock you choose in the morning, okay? But if you have this kind of funny, fancy thought, it's not the same. You cannot put one in your left one, in your left feet or the right one, okay? So this is more or less the interaction. Of course, this interaction, if we go to a smaller system like this chiral molecule, okay, if you have the right one and the left one, and you smell, and because we are made by these chiral receptors, okay, at the end, for example, this one, you will smell like orange, okay, and this one smell like lemons. So the interaction is completely different, okay? So it's very important because now people is doing self-assembly, okay? And it's using, for example, with these nanoparticles, the recognition of these systems, and it's a very, very specific because these DNA links, okay, recognize in some way and in some interaction that you can, and the people can do now a lot of different supracrystals that are called, done by these nanoparticles. And, but for example, there are systems which are also intrinsically chiral at the nanoscale, okay? For example, the nanotubes, which are denoted by these two indices, I'm going to explain why these two indices. Okay, for example, if you have a nanotube with an N index, only the zigzag, which is the N comma 0, and the unchaired one, the NN, are a chiral. Okay? However, once you give an n number, you give a radius of the nanotube, and at the end you will have a lot of nanotubes in between which are chiral. For example, if you have this small index n, you have a total of 5 isomers, okay? And 3 of them are chiral. Only 2 are not, the zigzag and the archer. So at the end, you have an index n equal 18, for example, you have a total of 19 enantiomers and 17. That means that most of them, are chiral isomers. The same happens with fullerene, except with the C60. Okay? If you have higher fullerenes, for example, let me just go here. Okay? You will have, for example, in the in the fullerene with 100 atoms. Okay? You will have a total of 450 enantiomers, isomers, sorry. And at the end, you will finish with that 412, which are chiral. Okay? So also nanostructures are chiral. And the point is that there are a lot of things in the literature that people want to use nanotubes to drug delivery, but the drug delivery wants to act with those are chiral, and if they are a vehicle to go and to apply the drug, well, they will interact with us, and we are chiral, so we have some interaction. We could have some interaction depending on the chirality of the carbon nanotube, as well as, for example, well, these are two different examples. Okay, so and the other way we can use it in a good way, in the sense that we can, if we understand this kind of interaction, we can have this optically active system, like this small gold cluster with some ligands which are not chiral, but because of the interaction of the ligands and the gold, these systems at the end are chiral. And you have these properties that usually metallic nanoparticles have, like optics for sensing, for electronics, for molecular recognition, labeling, and anti-separation, asymmetrical policies. Okay, at the end, you can find that you can have these chiral plasmonics, okay, chiral nanoplasmonics, some specificity. If you understand this interaction, you can handle in the electronics, for example, to have a spintronics. You can handle at the end the spin of the electron spin, for cell assembly, as I showed, specific labeling. And in this case, probably we can understand why homochirality in living beings is the origin of life or whatever. I cannot say more about that because it's some kind of philosophical kind of things. But it's very important also in pharmacology. Okay? So chirality nanoscience means that you can generate chiral nanostructures. You can use for synthetic separation, molecular recognition, and so on. So let me just go a little bit to the theory. I'm not going to show a lot of things about theory, but just to do the work. Okay, so we have this linear polarization of light with the left and right circular polarized light. Okay, then it goes to this chiral system, like these fullerenes, these chiral fullerenes, and at the end you preferentially finish with that it absorbs more this blue light, circular blue light than the red one, and you finish with this. So at the end you have the left field, electric field is given by this, where this is the molar extinction, okay? L is the size, the path of the cell, the length of that the light go, and lambda, of course, is the wavelength of light, and this is the right electrical polarization at the end. So at the end, this, the angle of this of the electricity, okay, is given by this relation, okay, and usually these angles are very, very small, are some milli-degrees, okay, so we can approximate this like this equation. Here one parenthesis is missing, I don't know why, and I always do the same, no, I did not correct. Well, and you finish that at the end the electricity is given by the difference of the molar extinction, okay, of the left minus the right one, okay? This is the definition of circular dichotomies. And of course you can do several things to relate with other quantities. Now what we have done? Well, we start, for example, for a system, in order to understand these interactions between molecules and nanoscale systems. Well, we start with a Hamiltonian, okay, of the basis level of the system. Then we perturb the system with light, okay. This perturbation we rewrite in terms of a multipolar series of multipolar contributions that have to do with the electric dipole, the electric monopole, the magnetic dipole, and so on, and we solve all the terms. At the end, we finish that the molar extinction is proportional, for example, to this term, which is the one that we usually have, which is the light scattering, that is responsible for the light scattering, absorption, refraction, all the linear things. Then we have a second term that has to do with something that is called the natural optical rotation. And this is the parallel effect. For example, if you have a system and you have an external magnetic field, you will see optical activity because of this. Or if you have an ordered system, an isotropic system but ordered, you will also have this term that you will see this natural optical rotation. And finally, you have this term, okay, that is related directly with the circular dichotomies that I showed. So at the end, the response, okay, for example, the light scattering is more than 100 times larger than the effect to measure the circular dichotomies if you want to measure the difference between the two. So what happens when you go with the left-handed and right-handed light polarization and you want to measure this symmetry inversion in 3D systems, in three-dimensional systems, to see if the chirality of the system, it's like you replace, for example, in these terms, the x, y, and z by minus x, y, minus y, and minus z. And this is called the parity operator, no? In physics. And this is accomplished when we consider the left-handed and the right-handed circular polarized light. So at the end, if we operate this parity operation in the first term that gives us the polarizability, we find that the left one and the right-handed one are equal, okay? For the second one, for the natural optical rotation, it happens the same, it's exactly the same, it's the same. However, the third term, the left one is equal to minus the right one. So when you do the circular dichotomy, okay, so the response of the left minus the right one, you will finish that this is proportional only to this term, okay? Only to this one. And this is the way that we can relate the macroscopic response that we measure in the labs with the microscopic expression or quantum mechanical expressions for the circular dichotomy. So this term is given by these matrix elements, which are related with the electric and the magnetic dipole operators, okay? And so we will finish with this expression, and I will finish here this theoretical part. So what we did is to implement this in a code, in an initial code, and we did a lot of things. because, for example, to study the system, well, you need the precise knowledge of the atomic configuration. You need a first principle method in order to quantify a lot of properties of the structure, like the electronic, to do a structural optimization, a structural electronic calculations, and these optical properties. And some methods are available for very small system. The coupled cluster method is very, very accurate, but you cannot go for more than 10 atoms, for example. Or you can use also the time-dependent density functional theory, but this is still very expensive computationally speaking. So you cannot study these nanoscale materials with that. So we did, of course, calculation with Fullerene. We compared here our results with the experiments and with other theory. Here the same for different Fullerenes. These again our results, and these are the experiments, and these are the, for example, the time-dependent DFT that is, according to us, our method is much better than time-dependent DFT, because time-dependent DFT you cannot be very accurate if you have these very large systems. This has 84 atoms of carbon. Well, let me go a little bit to these nanotubes. And as I show you, well, the single wall carbon nanotubes can resemble like graphene or roll up, no? A graphene shape, no? So, for example, you can have this part of the graphene, and you can go and to have this kind of nanotube, which is a chiral, the armchair, okay? Or, for example, you can choose another part of the graphene, and then you will have this, and at the end you will see that you have a chiral system that goes like a helical or twisted thing, okay? Okay. To construct the nanotubes, for example, we start, as I show you, the zigzag is like to have, if we have this hexagonal lattice, for example, we have two vectors, A1 and A2, and you can construct by this, and that means that you have 5 A1 vectors, 1, 2, 3, 4, 5, and 2 vectors, and at the end you construct a unit cell that you go to infinity and to have a very large system, okay? And you can have this, the phi 2, for example, and the diameter of the nanotube is given by this one, no? This vector. So once you have this vector, you can start here, for example, in this zigzag, the carbon nanotube, but you can have a lot of carbon nanotubes with more or less the same diameter, okay, of the tube. And for example, you have this one with the A2, etc., the 6,5. But if you go further to this one, after another 30 degrees, you will recover the zigzag. However, in that case, you will finish, for example, with with an enantiomer, okay, the phi 2 or the 2,5. Okay, you will have the mirror image if you do in the other way. Okay, and they have different properties. So these are the two mirror image related chiral nanotubes, and of course, they were known since 1991. Okay, and of course, the proportion is racemic. Okay, but until 2007, the people could separate them, okay? So they start, for example, with a solution of a mixture of different diameters, then encapsulate or wrap the system with DNA or with another kind of chiral surfactants, like diporphins, pelicinamine, and so many others, okay? Then they do a method which is the density gradient centrifugation that separates with the specific mass of the system, okay? So they could separate by the diameter, and also if, for example, the molecules interacted with the nanotube, with one of the nanotubes or not, okay? And finally, they could separate one enantiomer from the other, the NN from the MN, and they characterize this by using the circular libraries, okay? However, in the small carbon center, the people know that if the light rotates to the left, they call the left enantiomer, the right enantiomer. So there is no way to know which enantiomer you have. Okay? In circular dichroids, you have the same response in the spectrum, but the contrary or the opposite sign for one enantiomer and the other one. Okay? So, which are the chiral surfactants that are interacting with which nanotubes, the people doesn't know very well, how this enrichment can achieve. So it's not easy to know, and this is why ab initio method is important, because if you want to know what is named in chemistry the absolute configuration, you need to compare always the circular dichroids, the measured circular dichroids with the calculations, okay, with the theory. There is no way to know for larger systems if you have the left-handed or right-handed, and you know the correct reaction of these chiral systems. So we have these two enantiomers, the 6,4 and the 4,6, for example, and of course the intensity here we don't have millidegrees because we need the concentration of the system and so on. However, if you consider, for example, the enantiomeric excess, that means that probably not all the molecules interacted and wrapped one of the nanotubes, and you at the end finish, for example, with one enantiomer, you will have, for example, in the best case, well, the 100% of the 6,4 nanotubes and the 4,6, but if you have a less successful method to separate, well, you can have a very little or very small intensity in the circular dichroids spectrum. So CD detects very, very small differences. Just remember that the chemical composition is exactly the same, just the geometrical part that one is the mirror image of the other, but it can be, for example, very small, and you would like, for example, to enhance this signal. Okay? So this is the comparison of our results with a nanotube. This nanotube is the 4,6. So in this case, for example, these people use the DNA to wrap the system, and what they are recovering in this case is the left-handed, that we can call the left-handed, the 4,6 nanotube instead of the right one. Okay? So we did a lot of systems, the 6,4, as I showed you. Also, this compares very well with other separations, with other experiments. And again, they found with a different system, with a different chiral surfactant, the sodium, I don't know how this pronounced in English, cholate, okay, thank you. And they also finished with the M enantiomer or the 4,6 system. For this, which is also a small one, again, for example, they use the porphyrins, and again finish with this left-handed system. But if you go to larger or diameters, like the A4, the first ones that I told you are here, but now these are here. This is not very well, okay? But if you go to larger ones, like the A4, A3, and 7,3, what happens is that now they finish with for example, with the right hand, okay? And so that it seems that not only depends on the interaction of the chiral system, but also on the diameter of the nanotubes, okay? And that can be because of the proper molecule can be larger or smaller okay, and can interact in different way or grab the nanotubes in different ways. Now, let me just talk a little bit about these nanoparticles. For example, for many years, people have used ligands in order to have a very, very well-defined number of clusters, atomic clusters, with a specific number of, for example, of ball systems, okay? And they use usually these glutathione ligands to achieve this, so they can separate again with very, very specific number of atoms. So this a chance to have this specific atomic configuration. But some time ago, they observed that, for example, these small systems with the glutathione, the glutathione, for example, exhibit circular dichroism above 5 electron volts, okay? But the complete system, the glutathione with the nanoparticle, with the metallic cluster, show, of course, a larger intensity, quite different from the one from the glutathione itself. And also there are some response in energies much above the one of the glutathione. So something is happening, okay? And one explanation is that both clusters can be inherently chiral or can be chiral after the interaction between the ligands. And Ignacio Garzón has done these calculations for the atomic configurations, and well, we have collaborated in that way to try to find what is the specific relation. But let me go to another system. This is now silver. And for example, as I told you, the CD could be very small. And here they have a left remaining molecule alone, which is this line, the black line, this one, that I have here. The response is here. And once the molecule is in contact, for example, with different nanoparticles, this is a small one, okay? For example, here, the response change, okay, inside, and some other features, some other peaks appears here. And here, for example, is where the plasmon of this very large nanoparticle, silver nanoparticle is. And now if you compare the 100 times the molecule interaction from here, okay, and this one with a nanoparticle, you can enhance in some way the circular dichroism, okay? However, this is not easy to understand because, for example, in this SER, surface enhanced Raman scattering, this depends on the electric field, enhancement of the electric field. But here, you have the interaction between the magnetic dipole and the electric dipole. So it's not easy in this way to understand why you will have this kind of enhancement. And for example, if you compare one, or you take one of these peaks at a given wavelength, you can see that after some concentration of molecules, it doesn't enhance anymore, okay? It's very, very homogeneous. It reaches some kind of intensity. Or, for example, it happens also over the time, no? That at the beginning you have a large enhancement and then you don't see anymore. And so this is a very complex problem, okay? Why? Because it depends on the kind of the molecule, the chirality of the molecule, the number of absorbed molecules, the absorption side of the molecule's orientation, the molecule arrangement, no? The arrangement of the molecules can be also chiral. For example, the chirality of the nanoparticle, the size of the nanoparticle, and the atomic species of the nanoparticle. So this is a quite complex problem, and we have done some calculations in this way to try to understand. So we start, for example, with this chiral cluster, a very, very very symmetric one with 55 silver atoms, and we start with an achiral molecule, just to try to understand what is going on here. And so we represent this with this schematic. It has a 5-fold symmetry, this one. However, if you only have one molecule absorbed, now, for example, all the 5-fold symmetry disappear except this one. You only have only one symmetry. But now if you rotate, for example, the orientation of the molecule, you are breaking again the symmetry of the system, and you can see that you can control the interaction, and you can control the intensity of the circular likewise. Okay, so by changing the orientation of the molecule, for example, here it's close to 0, this white line, but this one, which is 36 degrees, you can rotate in one direction or the other one, so you will have like a left-handed and right-handed enantiomer. You have the same, but with an opposite direction. So we can induce optical activity in that way. Okay. We did the same with, for example, with the gold cluster, but in this case, by looking at the atomic position, we can see that the gold cluster distorts. Okay, and this is just because the d electrons are close to the Fermi level. There is this spin orbit interaction, this relativistic effect that makes that gold can move very easily as compared with silver. Okay, and for example here, what we can see is that we have from silver a small circular dichromate, okay, in the same system, for example, and if we just look the contribution just for the metallic part, we see that this is almost 0. However, if we do the same with gold, okay, this is just one molecule, we find a much larger circular dichroism, this one, okay, and we can see that if we also take again the contribution from the metallic part, we see that most of the contribution comes from that metallic part, okay? So what is going here is that the distortion of the gold nanoparticle is given a more intense circular dichroism than just to break the symmetry with the molecules. Now, what we did is again the same, but what we did is just to decorate the 1 and 2, which are the chiral ones, because we always recover one plane of symmetry, and we compare, and well, we have a very small intensity in the circular dichroism with D. But now what we did is just to break the symmetry with a third molecule in the left-handed and the right-handed one. And what we can see is that now we have a response which is 40 times larger. Okay, so to have an arrangement, a chiral arrangement of ligands that induce again circular dichroism, and this is induced in this part of the spectra, which are for small energies, as the one that, for example, the one that, for example, in the first experiments observed, that appears where the molecule doesn't have any. This is an achiral molecule, so it has not any circular dichroism. And of course, if you do the racemic mixture, you recover that you don't have anything, no? As expected. Now, we call this an induced optical activity by the arrangement of the ligand. And we did the same, but now by using chiral ligands. This is the cysteine, this is the circular dichroism coming from the cysteine. We did the same with 1 and 2 ligands in the same way that we decorated the previous slide. For example, with the chiral molecule, what we found is that now we have a response which is an order magnitude larger. So the response of here, we can identify a little bit with the left cysteine. Again, we do the left-handed and the right-handed system. Okay, when we do that, again we will recover this induced chirality just by the arrangement. But now, if you look here, we don't have the same response, but with opposite signs. What we have is something that we can understand very well, but when we do the racemic mixture of the left-handed and the right-handed, what we find is now a very clear response here. And this response, for example, is more or less proportional to the number of ligands in the system. So it seems that if you have 3 ligands in the system, three cysteines here, you will have and enhance the number, the response, for example, of the molecule. Okay? So we have now the intrinsic chirality of the molecule. And it is enhanced, no? Again, because the intensity, if you look at the beginning of the left cysteine, it was just like 5, and now it's around 45, 50 times, 10 times larger. Okay? We do the same with the induced, the intrinsic, with this chiral arrangement, and in all the time we find this proportionality with the ligands. And well, we obtain this. I don't have talked so many times, but just let me show you this last system that we have. Well, we have two graphene layers, okay, with a very atomic state control that can expand, you can have a very uniform film. And now we have, for example, contribution, collaboration with this group in Cornell, Gibbon Park, that have moved right now to University of Chicago. And for example, they can do these systems to put one of the graphenes one over the other, and then rotate. Okay? And then again, discover that if you rotate to the left or to the right, okay, you have the same signal but of contrary sign. And why not? Because if I can see this here or this, okay, but if I look in the other side around, why is that? You know? Why I can change, for example, the magnetic dipole or the electric dipole direction just by rotating one direction or the other. And well, they found, for example, that they do this for so many angles. These are just some examples. And one interesting thing is that in some sense they can manage the chirality of the system by the angle and the energy of the system. So you can have a very uniform and very large system where you can manage the chirality, where you can, for example, manage the interaction between the chiral ligands or chiral molecules, etc. Okay? And the point is that, for example, in graphene, any asymmetry, okay, has an effect to generate a pseudo-magnetic type, okay? This is very well known, okay? And for example, if you rotate in one direction or the other one, to change this magnetic dipole, pseudo-magnetic dipole, just by the interaction of the two sheets. This is very well known that if you stretch the graphene, or you put the graphene over a boron nitride film, you can change for example, the current, the direction of the current of the electrons. So you can change this magnetic dipole, and this is why you can handle very easily in this way, in this system, in this particular system, which is not in any system, in any other. And well, we did some calculations, and we, in this particular system, arrived that the circular dipole, or this term which is related to the circular, is directly proportional, this is a constant, to this momentum, that means to the current of the system. So you can manage this quantity. Well, this is the explanation, the theory, and so on. I don't have so many time, but just let me just to say that these kind of systems, which are very large, you can manage the chiral interaction or the specific chirality of the system, can be very important for the chiral separation of molecules, which is very important in the pharmaceutical industry. Or, for example, you can do spintronics. Okay? In this case, for example, you can not only have the current, the charge current in a system, but since you have a very specific interaction with this pseudomagnetic moment that you can create between the two graphene, the rotation of the two graphene sheets, you can manage, for example, the alignment of the spin, and at the end you will have what is named spintronics. And let me just finish here. Okay, this is some of the people that I work, and well, the results that I presented are mainly from Francisco Hidalgo, who has just came back from a postdoc in Barcelona. Now he's here. He came back this month. And Ariadna Sanchez, who was my graduate student also, and she is now in the Autonomous University of Hidalgo in Apan. So if someone wants to recover her from Apan, it's just pulque, and even not pulque anymore in Apan. Okay, and well, it was supported by many agencies, and thank you very much for your attention.
Thank you for the very nice talk. I just want to ask you something to see if I understood correctly. When you talk about this the parity operator, and you talk about this G tensor, this second order tensor, and then you said that the parity operator changed the sign of this G, right?
No, if you apply that means the parity operator is like you change
the sign.
Yeah.
And then you have products of the dipole and the magnetic dipole, right?
Yes.
And then you change when you do the parity operator, you change the sign of the dipole, and then the sign of the magnetic dipole remains the same. And
you can have both, no? What you need at the end is that, for example, if they are transversal, you don't have any sign. Okay? So to have the largest signal, you need to be parallel or anti-parallel, whatever, to have the largest one. And for example, if you look the magnetic dipole is given by, if you look given by the current of the electrons, so it's an operator that you can write like the cross product between the position and the momentum.
It's given by the angular momentum.
Yes.
I was wondering that when I do the parity operator, the angular momentum doesn't change because I have products of coordinates and momentum, and then I have minus.
Well, in that case, the dipole, electric dipole changes.
Is the one that changes.
The one that
changes. The dipole changes and the magnetic dipole remains the same. Okay, thank you.
It's a game between the two. Yes.
Thank you.
Very interesting work. I was wondering if you know about the assignment of these electronic transitions that you are observing on these materials, because the match that you get between experimental and theoretical in the energy axis is pretty amusing. So that means you really know what each transition.
Okay. Well, you mean because I'm using DFT? Yeah. Okay.
And how will this compare to TDDFT, for example?
Yes, sure. Yes. For example, here, this system doesn't show any tectonic effects. This is a good thing. And what we just do is just to open the gap in order to match the gap of the experiment. And it's the only thing that we do. If you have excitonic effects, you can have another kind of things, and then you will need time-dependent DSP or GW approximation, whatever, any other kind of thing. But in these cases, fortunately for me, they doesn't have this kind of interactions, so you can just open the gap and the matches with the experimental are very, very well. That's it. You can write it. Thank you very much. Tengo que bajar.
Second talk this morning will be given by Professor Alfredo Alexander Katz. As Professor Nogues, he also graduated from the Faculty of Sciences of this university. At present, Professor Alexander Katz holds an academic position as researcher at the MIT Department of Material Science and Engineering. His research is mainly focused on the self-assembly and dynamics of soft materials of novel applications in fields such as medicine, biology, physics, and chemistry. Professor Katz, thank you for speaking today.
Muchísimas gracias por la invitación. Es realmente un placer y un honor regresar a mi alma mater. Y bueno, qué bueno que es con una celebración de los 100 años de la Facultad de Química. Yo no estudié aquí en Química, pero definitivamente celebré mucho en las quemas de bata. Que ya escuché, ya no se hacen aquí enfrente, pero bueno, es realmente un placer y les voy a hablar un poco de lo que hago en mi trabajo. Now I'm going to change to English, and so I work on the field of soft matter, all this squishy stuff that actually makes us, so we're all made out of soft materials, and believe it or not, the new jets are all made out of soft materials. Not all, but 50% is plastic of the new jets. So, ¿se apagó? Can you hear me? Hello? Oh. Okay. So, and I think we should be very proud of that because it all started here about. Yeah, it all started here. Is that okay? Or I can lower it a little bit more. Bájale. So it all started here, actually with the hule, you know, the couch, you know, the rubber, and in fact, actually, the Aztecs already knew how to do what Goodyear discovered many years later, which was the vulcanization. They used some different acids to do that, but they were already doing that many years ago. So from that point on, now we have plastics, and we cannot live without plastics. And all of those are actually what today we call van der Waals materials. They're all bonded by van der Waals forces, and some other chemistries. But you have to make them, and that's where a lot of chemistry comes in. So anyways, what I was going to talk to you today was about, I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to you today was about, what I was going to talk to basically the synergy of the synthetic world and the biological world. And in a very specific case is about how nanoparticles, so I'm going to give the other side of nanoparticles. I'm not going to be talking about electrons or anything like that. So I'm going a little bit up in scale, and how these objects actually interact with biological membranes. So let me tell you why this will be important. Okay, so let me tell you why this is important. So imagine you're in the lab, you're preparing this drug delivery particle, let's say it's this, and you take your effort in making the surface, you put it into the body, and suddenly it gets covered in all these proteins. That's called a protein corona. And what happens? Well, this is now a dress particle. And this is not a quantum dress particle, this is actually a classical dress particle. But now you have different chemistry on the surface. It's not the chemistry that you put, it's the chemistry that basically biology took and put on top of that. And there's been a lot of effort to try to understand how to control that chemistry and be able to control what absorbs and what doesn't absorb. And in particular, it will be nice to have things that do not absorb. So what I will be talking about is actually a class of particles that don't, at least in the blood, do not absorb too much. But this is one of the biggest challenges today in biotechnology. And sometimes what happens is that they bind to stuff that you don't want them to bind. And that can be actually pretty toxic. So if we actually are able to do this, then essentially it opens a huge door of applications. So you can do very targeted delivery, and at the same time you can actually image very locally some particular aspects of your body, or even if you take it outside and you don't want to do that, but you want to template inorganic materials, it will be actually very important to know how to interface these two worlds. So the particular thing that I'm going to be talking about is interfacing with biological membranes. So I took this picture from Wikipedia, and so the membrane of your cells is composed of 50% of phospholipids, or these guys which have two tails, and then the rest is essentially proteins, and there's some sugars coming out here. Some of them have more than others, and so forth. And there's something very peculiar about it, and it's that the head groups are polar, and they actually like water a lot. And the groups that are here in the center, they're hydrophobic. Okay, so they're carbon chains, alkane chains, and or non-saturated and saturated. And actually this becomes a barrier. So it doesn't let ions go from one side to the other. So let's say that's outside the cell, that's inside the cell. So ions don't cross this. To say don't cross them, they don't cross them on the short time scales. They cross probably in a day or so. They would actually start. They will take to cross this membrane. So this is also taken from Wikipedia. So they're impermeable to ions and polar molecules. And this has actually been measured. I don't think we have too many experimentalists on the audience, but if you take a vesicle, which is just this without the proteins, you make it a shell, you osmotically shock it, either you compress it or you expand it, and the ions, the salt is not going anywhere. It's staying there. So that's an important characteristic. So essentially all the transport that you get across the membrane is controlled, let's say, by channels. And one interesting aspect that you might be asking, because you will have proteins that were made inside but come outside, and they have charge groups, how do they do those? Well, you have special proteins, which is probably not this one, but you have this called translocanes, where you do the synthesis in the membrane, and then it comes out, and it's already basically spanning the whole membrane. So what I'm going to be talking is, well, what happens now when I take a nanoparticle like this and I put it in contact with one of these violators? And I'll tell you why I'm interested in this problem. So typically, if you take a nanoparticle and it's charged, most of the particles are positively charged. If you do that, it makes a hole. And you'll see, oh yeah, it gets into the cell, and but it makes hole, and it's very toxic. You don't want things that are too toxic for cells, especially if you want to do treatments. But
And by the way, all these amyloids, so all the bad things that happen, prions and so forth, they're typically positively charged, typically, not always, but the interesting part is the small fragments that are the toxic. It's not once you see the big fibers, but you're done. But the small ones are the ones that are toxic and are making holes basically on your neurons. So they're depolarizing your cells, depolarizing your neurons, and then you cannot, basically doesn't work. So we were very interested in seeing a different class of nanoparticles. So it's a nanoparticle, and also has ligands, just like Cecilia showed. These are different ligands. They're not glutathione. These are just carbon chains with some sulfonate. I'll show you in a second more what they look like. And these are negatively charged, and we want to understand what happens with those. And the reason why we wanted to understand is because of these results that happened in 2008. I got a chance to see this right before publication, but it's very simple. So they take these particles, they have either completely charged, so those ligands are all sulfonates, or they mix them with hydrophobic and hydrophilic groups, so some have sulfonates, some don't have. And what they see is that these particles go into the cells. And one of the things that they saw is that there was no holes being made, because they can check this with another fluorescent dye that's in the suspension. And another sign that they could see this is that if you see these particles, which are in red here, they're distributed somewhat homogeneously over the whole cell except the nucleus, which is that dark center. And normally, when you take things from the outside in this endocytotic way, and I'll show you in a second what endocytosis is, it looks very punctured. But one way to actually kill those energy-dependent mechanisms is to cool it down just before freezing, and they still saw that these particles were coming in. So, and in that case, we know that it's not coming through this endocytotic way, which is when you wrap these particles and then you bring them into the cell. So there's something funky going on here, right? I just told you membranes are impermeable to ions, and then I'm telling you, or they were telling me, that they can get 100 charges or more from one side to the other. And it's not taking weeks, it's taking minutes, okay? So, so that was very puzzling. And I didn't have a good answer, but I said, okay, this is a very interesting question. So we started to think about what could actually be happening here. So one of the interesting observations was that composition, so what is the chemistry on that surface, actually matters. So you take this branch ligands, it doesn't work as well as it works with this non-branch ligands. And that's something that was a good indication that there's something about packing and so forth in these ligand shells that might be important. So, so these are the particles. They are a gold core, and through thiochemistry, they bind these ligands, mostly carbon. So these are 11 carbons and one sulfonate group, and these ones are 8 carbons. So this is how they look when you put them in atoms, and this is the chemical diagram. So one of the things that I want you to remember here, and we call them amphiphilic nanoparticles, is because while they're charged, and these guys are really soluble, I have them in the lab, they last years, no aggregation. Still, if you look at it, it has a lot of hydrophobic material in it, okay? And here's where I say the magic comes. So this is soft. They can rearrange, okay? So there's ways that you can actually change the local structure of that interface and actually start making it hydrophobic or hydrophilic or both, in this case amphiphilic, in response to different stimuli. So it's actually very interesting. So this is normally the nightmare of chemists, the ones that make the particles, so the particuleros, but not the physics, but the chemists. So which ligands you use to make some particles of a specific size. So we think this is the magic for this particular ones. And so at that time, I wasn't convinced, and so we have students here, right? So I said, well, this is very risky. It goes against the first 3 pages of any biology book. So I'm not going to put a grad student. I put an undergrad student. So, estudiante de licenciatura. And he said, look, if you solve this mystery, I'll invite you to the most expensive restaurant in Boston. And my wife was there, and she's like, what the hell are you doing? I'm like, well, okay, we'll see what happens. So he actually stayed with me for his PhD, and we actually solved the mystery, as you will see. But it was complex, right? So this is a complicated question. So the first thing we asked is, well, can we do this in without biology? You know, I don't want to have all these proteins and so forth. So let's just do it with vesicles. And these are the results with vesicles. So these are negatively charged vesicles. They have two types of lipids. They have DOPC and DOPS. It's about 20% of DOPS. And when you don't have salt, the electrostatic interactions are very strong, and they prevent actually the particles from getting to the surface. But at the moment you add salt, what we start seeing is that while this dye, this is in green, doesn't go into the vesicles, these particles start going into the vesicles, but then they didn't go everywhere. They just went to the membranes, and we can see the membranes from our DIC. And they went to the internal membranes as well. And just keep this in mind, they went to the internal membranes as well. So, so we say, okay, so this sounds that it's something with the lipids, and we're going to start studying how are these lipid nanoparticle interactions. And so the model that we were working was, if you're going to go through this bilayer, or if you're going to actually go through the bilayer and not make a hole, you need to fuse with it. There's no chance of going through without making a hole unless you actually become part of this membrane. And to become part of this membrane, what you have to do is you have to change your effective surface chemistry and create what we call these hydrophobic bands, okay, that match the bilayer as they're coming in. So this is something that membrane biologists call matching, okay? So you're matching, you're hydrophobic matching this bilayer. And you will put all your charges on one side or the other side where there is actually water. Okay, so there's two options to do this, and we've done both, but I'm going to only talk to you about one because this is the predominant method, the predominant pathway that we think happens at least for a short time. So one way is your ligands are, just floating on the surface of this gold particle, and they can move around. They can all reorganize as you're coming in contact with this bilayer, and it puts all the hydrophobic ligands to the side of the hydrophobic tails, and you leave all the charged ligands on either side. The other way is to do what biologists call snorkeling. So if you take proteins, sometimes they actually have proteins that are in the membrane. Sometimes they have groups, amino acid groups that are actually charged. And these groups try to go to the surface to get solvated. And this going out, which I call the anti-snorkeling because they're going for water, they're not going for air. It's basically this ligand that was in this point, it cannot move from there, but it will try to reach the surface, okay? And it will try to become solvated. So, and the other ones will come here, and you will still have this hydrophobic band exposed. So we've done both because there's a lot of divergence in the literature of how fast these ligands move. But even with that, we think that this is the right mechanism, at least for the first part, which is mostly what I'm going to be talking to you about. Okay, so how do they get into these bilayers, okay? And I'm going to tell you, so the first part I'm going to tell you how that happens. And in the second part, I'm going to tell you, on what ranges this will happen. So that will be more of the thermodynamics. Like if I wait infinite amount of time, what is basically the thermodynamics of the system, and which ones are actually going to go into membranes and which ones are not. And at the end, I'll tell you a little bit of a summary and analogy. Okay, so this is a movie. And this is what how a nanoparticle dances on the surface of a bilayer. So there's the nanoparticle, and it's just happily dancing on the surface of the bilayer. So what keeps it on the surface of this bilayer is a charge dipole interaction. Okay, so there's dipoles on the surface, and you have this charge group, and this particle will stay here. This will go for 500 nanoseconds, and it will just stay floating around in this bilayer, and we would actually not see anything. But in order to understand what can be happening here, and and where are the points where we can actually think these could insert into this bilayer. What we created was some defects. So what we created is something called a ribbon. And if you create a ribbon, what you have is a flat part of this bilayer, and on the borders, you just break it. So you basically just break this bilayer. So it's not continuous. So lipids have two tails like this. So let me think all these lipids are like this. Okay, so they're happy. They're like liquid crystals. They're happy in this conformation. They're happy on the conformation on the bottom. But as they go from one to the other, there has to be defects being formed because the packing doesn't work as you're going around this curve. And what that does is that if you see this red, it means it has the tails, its hydrophobic tails, have much more contact with water. Okay? And originally we were thinking it's all the nanoparticle, but actually it's not the nanoparticle, it's the lipids that control this interaction. So if I put this particle here, I will have to wait many, many times. It does happen, I can tell you, it has been proven, but one will have to wait a long time. Now, if you do it on the side, it's almost not instantaneous, but it happens pretty rapidly. And one interesting aspect is that this actually gave us the mechanism of how it happens. If I didn't show you this particle in different colors here, you wouldn't even know there's a particle inside. So it is really like a chameleon. It just became part of the membrane. It has its charge on one side, on the other, and it's happily living in that membrane. So these are some experimental results actually on that. These are defectors membranes, so these are all the edges. And when you see this brighter thing, those are actually the particles. And they're attacking all these defects basically from the side. So the nice thing in computation is that then we can chop our simulation, say, oh, it went in. So we have two basins. We have a basin that has that you're outside. We have another basin, you're inside, and we think there's a barrier between them because we don't see them go in immediately in the other cases as well. So let's see what are those barriers. So so basically here the particle is staying outside. There's a very rapid process. Something happens here that rapidly gets into the bilayer and then slowly diffuses into the center of the bilayer. So what we use is committer analysis. It's not a very fancy technique, but when you have no clue about what are the reaction coordinates, is one that you don't have to know exactly what happens. And so basically what it does is you chop your movie into pieces of time. So this is a long time. So there's a reaction coordinate, but this movie is not going along your reaction coordinate. It's going a long time. Okay? So you chop it into very little pieces. So perhaps you're getting close to the transition state. So on time you go up, and then you go away from the transition state. You will go down, and so forth. And what you do is you chop it, and then you launch. In this case, we launch 40 simulations from each of those points. And we ask how many are going forward, meaning how many are going into the bilayer, and how many are actually going out from the bilayer. And when that becomes 50-50, when it's 50% of the chance going in versus 50% of the chance staying outside, that's our transition state. So, so this is the committer analysis P, this black thing. And when it crosses 0.5, that's when we know now it's going to go into the bilayer. And interestingly, it has to do with basically when more of these lipid tails are in contact with water, that's when it's much more convenient to make a contact with one of these ligands that are on the surface, which are hydrophobic. So you basically don't pay the price of the hydrophobic effect, and that's where it starts to get sucked. So we can go back to the movies. We know exactly at what time this transition happened, and there's two. So we've seen transitions which are called the elbow. So the lipids are, let's say, my arm, and one of the elbows comes out, so this gets exposed to water. If there's contact of that with your ligands, that will actually suck the particle in. And there are other ones called the splay. So now instead of doing like this, you have the splay protrusion, and this also will pull your particles in. So from those two mechanisms, what we were able to do now is say, well, okay, so, and these are all atom simulations. So what we're saying is like, okay, so let's think, if that's the pathway, I should be able to accelerate it by pulling one of these ligands from the flat bilayer, exposing it to water now. So now I don't have to wait for that time of this guy's coming out. That actually takes microseconds. So for all atom becomes complicated. And And then let's see what happens, right? Let's see what they do. And so this is, you can see the ligand right there. It's exposed. And what I'm going to show you is how these particles actually jump in. So in a matter of, you saw in nanoseconds, these things, they're like octopuses, like pulpos, right? They launch their tentacles in, one of those hydrophobic ones, and then they anchor, and they are there for a pretty long time. So this has actually fused, at least on one leaflet, with this lipid bilayer. And you can let it go for a long time. We went up to 150 nanoseconds. And this, for the most part, is the stable configuration, at least at the very beginning. But this is already fused. Okay, so it's exposing most of its hydrophobic content is exposed to the bilayer, and these ligands, as you can see, they're highly strained. They're trying to push out of this bilayer, they're trying to be solvated, and so you can see that they're all stretched, and I'm compressing this monolayer. So the next part is, well, how do I go from here to here, right? I want to go from there to the middle, because if I'm going to cross this, I have to go through the middle. So to go through the middle, what you need to do is you need to grab one of these charges and you need to cross. Okay? And again, bilayers are almost impermeable to charges. So that time should take a long time. It should take at least a day. But we're seeing experiments where this is happening, if not seconds, minutes. Okay? So what's going on? So what we went on is we took a single sulfonate group, it's a methyl sulfonate, and you take it out from solution, and then you drag it through the bilayer, and you calculate how much work you have to do to cross that bilayer. So that's going to be this black curve, and that black curve gives us the barrier to cross. We normally work, this is actually in kilocalories per mole, so it's actually a pretty high barrier. If we're thinking in thermal energies, that's even more. So it's very unlikely that this will pass. Now, if you take the same ligand and you now graft it, okay, so now I have the same sulfonate and I graft it and I drag it down through this, I obtain the red curve. And there's an offset of about, basically, this is about 10 kilocalories per mole, 8 kilocalories per mole, difference in the barriers, okay? And what that corresponds to is that in the rates at which these guys are crossing, it's a 5+ orders of magnitude on rates. So what was normally happening in days or weeks now happens in sub-second timescales. And that's why we're able to see this. And this is actually very interesting. It's very interesting for biology as well. So we've done this now with actual proteins and ligands and charged ligands. And what is very interesting is that this is a method to flip charges across the membrane in a fast scale, that you don't have to wait so long. So what is the key to that? Desolvation. The biggest price that you pay on this barrier is the solvation of the ions. So basically what you're doing here is you're partially desolvating those ions already in advance. So with this we have a complete landscape of how this process happens. Okay, so you start outside. These are the two pathways I'm showing you, but they both depend on the same. And now we've done it with coarse grain simulation, and even in flat bilayers without touching anything, you can get the same fusion. But you need, and it's also determined by this ligand. But you need those coarse grain simulations because you need to go to microsecond timescales. But anyway, so you have this attraction part. There's a basin here that's a dipole charge interaction. Then you have lipid protrusions, which is a barrier. This is of the order of 10 kVt at the most, typically 7. And eventually you have all these barriers to go into this state. And there's a bunch of things that you have to cross because you're flipping ligands. And they're of the order of 20 kT. So that's the biggest barrier that you'll find here. So now the question, let's say you have all the time that you want. So we're going to wait for equilibrium. What is the total energy gain of going from outside of the membrane into the membrane? Okay. So to do that, we couldn't do it with atomistic methods because we wanted actually to scan sizes, compositions, and so forth. So we went to a hybrid system which is the particle is in some sense atomistically resolved, but everything else is not. So the bilayer is just a field now and has an elastic component and it has a hydrophobic component. There's electrostatics, and the thing that costs more to calculate is this configurational entropy, and we use the Bennett acceptance ratio. So we basically are running like Monte Carlo on the system. So the driving force I was telling you is that basically you form this hydrophobic bands, okay? And basically all the water interactions with the ligands, which are in blue, are gone here. So now you don't pay this hydrophobic effect. And the bigger these particles are, there's more pressure because you're compressing more this monolayer, and these ligands will actually start to go into the bilayer, and they will pay a price because you cannot have charges in the bilayer. So we've done a bunch of these calculations. We've done it with different compositions. So 2 charge to 1 charge, all charge will be this all MUS, and the 1-to-1, which is 1 charge to 1 hydrophobic. And these are basically, whenever this is under 0, those are the values of the size of the core of the gold that will be allowed. So, so this one will go about up to 9 nanometers if it's 50-50. If it's 2-to-1, it goes about to 6 nanometers. And even very small ones that are all charged can actually go in. They're typically below 3 nanometers. We also put down a controversy of if the structure had to do something with it. These things are floppy. They're changing their conformation. So that didn't seem to give any difference. Interestingly, we can actually perform some experiments with our collaborators, and they look at what are the ranges that they see going in. So these are our simulation plots, and the ranges that they see going in are very similar to the ones that we have. And essentially the key parameter here is how strong is the hydrophobic effect. Okay? And if you have the right constant on that, which we use the one for transferring alkanes from water to an alkane solution, you get clearly good agreement with experiments. So I told you that, okay, so now I can fuse with this membrane, but what they were observing is that they go through the membrane, they don't make holes, and then they go in. But if you saw the scale there, it's about 200 kilocalories per mole. That's a very deep well. It's very hard to get out of it. Okay? So how do they get out? How do they come out? Well, there's different explanations. One could be there's actually glutathione in the internal part of the cell. There could be some exchange of these ligands, and then you do what I showed at the first slide, which is you change the chemistry of that surface, and then that actually goes into the cell. But what we think is actually happening is not that. And what we think is actually happening is that it's hopping from membrane to membrane. So I'm going to show you some cryo electron microscopy, and what you can see is that this nanoparticle is decorating many of the internal membranes of your cells. So you will see here, these are internal membranes, because your cells are full of membranes. They're full of membranes. They're full of endosomes. They're full of membranes that put the compartments for different functions and so forth. So, so we think that they're actually not just widely dispersed in the cell. They're actually in all these membrane structures. And this actually brings us back to, as I was showing you, these particles, they don't have to be cells. Now we can do it in vitro on a dead system. And they also go to the internal parts. So how is this happening? Well, it's happening by the same mechanism in which these particles went into the first membrane. So what I have here is now a particle that was in one membrane, but still some of those hydrophobic ligands were left here, okay? The moment another membrane comes close, it can now fuse with that. And now you have fusion of two membranes, and you can cross from one to the other. But it's always making these paths that are going through that membrane. Now, why this is important? Well, right now, in your brain, this is happening all the time, okay? Fusion of small synaptic vesicles need to go to the surface. They need to fuse with that surface, and then release those neurotransmitters. So that's done through a protein. It's called a snare. And this protein brings two parts together, and there's been a lot of discussion, but I think now it's starting to settle about what actually happens. And the moment the calcium potential goes up, this basically opens up, and then you get neurotransmitter. They fuse, and then you get neurotransmitter to be released, and that's basically how signals get passed from one neuron to neuron. So what I'm going to show you here is actually that these particles look or do the same function. These are synthetic particles, but they're doing the same function. There's a particle in that membrane. These are two separate endosomes. There's a particle right there. You'll see that it starts to fuse. It can form a fusion. We're not touching this, okay? This is just going. Now it forms a stable stalk, so that's called stalk. And what we're going to do now is we're going to mimic the action potential, so the input of calcium ions around here. And when you do that, basically you create tension. So you create tension on the outer membrane, and this will actually go into a full. Takes a little bit of time. There you go. Bam. Now 2 compartments are mixed. They have been fused. So, so this is actually quite interesting. It's understanding how to create things that make fusion is something that is very relevant, and I'll tell you some applications that this could be used. Okay, so now we have, instead of 2, we have 1, and it will keep on going. Okay, so if you want to see a different look, this is how it looks in the interior. This is the stalk. This is how it's forming. And now when you put the tension, there's tension and the 2 compartments just fuse together. Okay, so with this I'm gonna end, and I just want to summarize then what I told you, and I hope you got. So the first time is that I talked to you about this amphiphilic nanoparticles, and something very important from these particles is that they can reconfigure their surface chemistry. The reconfiguration of the surface chemistry is really interesting for its interactions with biology, and it can actually insert itself into membranes and just appear like part of your membrane. So while that gives us an opportunity, it's also a matter for concern, because all the nanomaterials that typically have ligands on their surface, and those ligands can actually be used, can be the driver that they can get into your cells. And finally, I told you about one application of this, which would be to understand this fusion process and perhaps do some regeneration, brain regeneration, because they induce fusion in parts where this protein wouldn't work. So, so probably you have never heard about snares, but when you have tetanus, you probably heard about that. This doesn't work, and then you become hard. Okay, so you cannot transmit signals. So I would just tell you a little bit about the future. So there's something very interesting for this. So we've been working quite a bit on drug delivery. They have very special properties. You can treat many interesting aspects. They also have this targeted ability that's still giving us questions. You can do sensing. You can do biophysics. You can even do things like desalination. So now we have a way to change, move water from one side to the other in a very fast timescale. And you could also do signal transduction. So you can have these things on the membrane, and then when you can now, it sort of hijack your cells, because if they're in the membrane, you can come from something on the outside that sticks to it and will now change its conformation and perhaps bring it out or bring it in, just as you do with proteins. So I'm going to just finalize with the acknowledgement. So Reed, this was the undergrad. He's now a professor in Wisconsin. He stayed with me for the PhD. And the person that's been doing the fusion is now Mukaram. Provani, Maria, and well, Provani did most of the experiments on this. And Maria and Randy, which are now, well, they were in EPFL. They did the experiments on the surface, and they synthesized a bunch of particles. And Sabrina has also been working quite a bit with us in MIT. So I've been very lucky to have Francesco Stellacci and Daryl Irvine as my collaborators. These are the funding sources, and we've made use of anything that we have available do the simulation. So I'm just going to thank you very much for your attention, and I'll leave you with a nice picture of something that doesn't happen when you don't have these nanoparticles, which is two kissing vesicles, and all these little lines that you see there are lipid channels connecting them. Thank you very much.
First, thank you very much, Doctor. My two short questions. The first one is, how is the dependence of the size, the relative size of nanoparticle and the thickness of the bilayer? Because in all your examples are very the same size. I mean, they are very close. But is there any difference if the nanoparticle is too small relative to the thickness, or very big?
Yeah, so there's an optimal, as I showed in the curves for the energy of binding in some sense. I'll show you here because here's the. So there's an optimum. So small is good. It still works, but the problem is that you don't have so much hydrophobic content on it. Okay, so you have fewer ligands. But as you go up, there's two competing effects. The more hydrophobic ligands you have that you can actually shield from water, better. But on the other hand, the bigger it is, the flatter this membrane is, and the more difficult it is to compress that monolayer to push it to the two sides. So depending on how much charge you have, so this is the composition. This is all charge, this is 2 charges per 1 non-charge, and this is 1-to-1. You can expand this to, double the size of the thickness of your bilayer. I don't think you can go much more beyond that, unless you start now changing completely the structure of that bilayer.
Yeah, exactly.
You had another question?
Yeah. My other question is it possible to freeze the process and to get a particle in the middle and not moving it from one side to the other?
That's a very good question. Yes, I think it's possible. But you need to do other chemistries. So we would need to have something hanging from that particle itself that stays only on one side. So this particle fuses, but then it has a big chunk outside, probably an antibody or some other protein, and that will hold it there.
Yeah, but that would be increasing the complexity of the structure.
Yeah, but okay, so I didn't tell you, but this has been actually an exploding field for my collaborators, because these things you can actually load with hydrophobic drugs, which are typically hard to take to cells. So they're like sponges. They suck hydrophobic stuff in this part. And if you target with an antibody, so normally they're taking one antibody to one drug. This, you take one antibody to 100 drugs. And you can come to wherever you want to go, and this fuses, releases its drugs into the bilayer, and then it partitions the way it has to partition. So it's not, it's worth pursuing. And for the signal transduction is where we want to do this. So if you have something on that outside that we can change its conformation, we can bring this in and out of the bilayer, and you can start changing those properties. There was another question over there. You have a question, right? Yep. She was.
Very nice talk. Congratulations. Just have one question about if you have tried another lipid bilayer composition, it's like DPPC or TOPS, but another lipid, or if you have also changed the thickness of the bilayer in the MD simulations?
That's a good point. So we've changed the lipid type. Things move accordingly. So there's only one important aspect. It has to be liquid. If it's in the gel phase, you cannot get into that bilayer. The gel phase is a frozen phase of the bilayer. But so if you're thinking of DPPC, if you're going at lower temperatures, you might actually start hitting the gel transition. But apart from that, it typically works. We've tried the charged lipids, like VOPS, as you're saying, and with the salt, for the most part, it actually works. But yeah.
Do you need any specific program like GROMACS or NAMD?
How do we do this? Okay, so so we do it at different scales, and so we've been using so we use the Gromos. It's not the last force field, but the one before that, and for that's for the all atom, and we use Martini polarizable water one. Martini is hard. I take those simulations with a grain of salt, and that's why we did the all atom first. But it has been actually confirmed by the Martini method. And there you can go to microsecond scale, so you just see it, right? So we didn't touch it. Boom, it just went and happened. But yeah, it's complex because for the students, right? So you have so in atomistic you have a cutoff of 1.2, and for some reason the Martini folks also said a cutoff of 1.2, but their particles are of 0.6. So when you're charged, okay, sometimes in the second shell you get a charge, sometimes you don't get a charge, so you start getting some very strong fluctuations on what actually is within your cutoff distance.
Thank you very much.
Alexander, that was really wonderful talk. I have two slightly technical questions. In the nanoparticles, the sulfur gold bonds are kind of labile, right?
Yeah.
So do you ever see, or but maybe it's not in the model, right? But do you see rearrangements because this thing can actually rearrange as a Janus particle, right? This thing can decide to put all the charges on one side and the other stuff on the other. Do you guys see anything like that? That's one question.
We fix them. We don't let them.
Okay.
We don't let them wait. Yeah. So and I think recently experimentally they claim that if you wait about a week, this actually can rearrange.
Okay. But it's
if you would leave it in that environment.
Right. And the second one is that there's evidence that at least for small molecules, when you go across the membrane, not at the end with the defects, right, but in a true bilayer, that you bring water with you.
Yes.
Right. That you actually make a little channel.
Yes.
But these things are too big, right? That doesn't happen.
They will bring water, and they bring water when these ones are, so they fuse in one leaflet.
Right.
Every time this is flipping, that brings water with it.
Okay.
Makes a water channel.
Yeah, and that reduces the barrier tremendously.
Yes.
Cool. Thanks.
¿Todo bien? ¿Qué?
Saber por qué, güey.
Oye, lo que yo quiero escuchar tu plática. Señor, ¿estás haciendo reacciones químicas como reaction diffusion y todas estas cosas? Cuando haces chemical network.
No, para que ya no voy a hablar sobre metabolismo. Hago más, redes. Este reacciona con este, y este reacciona con este. 70,000 reacciones con barreras. Y luego quiero hacer cinética de eso, y a lo mejor espacial sería padrísimo, pero...
Oye, lo que deberíamos hacerlo espacial.
Hagámoslo y hacemos Belousov-Zhabotinsky.
No, pero ¿sabes por qué? Vamos a hacer química topológica. Suena muy raro, pero cuando está hablando con este güey de Harvard Medical School...
Pasas topológicas de reacciones.
De reacciones, porque cuando te meten en el endosoma, cambió la topología del espacio. Porque ya no hay difusión de uno al otro. Entonces, estás cortando y puedes empezar a hacer cosas raras. Después puedes acoplarlo a curvatura, donde una dona no es lo mismo que una esfera.
¿Por qué no lo hacemos, güey?
Yo puedo calcular las raíces y luego
ya hay que hacer un proyecto juntos. Ya tenemos tenido, estamos juntos. Vamos a hacer unas cosas. Pero no voy a hablar de eso, le voy a decir a la gente que solo voy a hablar de termodinámica del metabolismo, porque ya mucho en la plática. Mira. Entonces lo estaba buscando, porque estás hablando de snorkeling. Dije, este güey habla de snorkeling.
Ahí les va un snorkel, cabrón. Por eso me perdí los últimos 8 minutos de tu plática. Pero bueno, ¿tengo audio o no? ¿Qué pedo? Espérame, ¿dónde está?
Pero el VGA, aquí hay este VGA y ahí está el...
Necesito el adaptador. Y voy al baño también. Si lo traes, pásamelo. Si te paso, sí te devuelvo todo. Es que los míos están, pero en una bolsita ahí que tengo. Sí, pero voy al baño primero, señor, espérame. Déjame darle la mano al desempleado.
Podré probar la mía, debería andar. Sí. Poder usarla así. No con esto, ahora, que también lo necesito. Se conectó.
Sí, hay una mutación en uno de los canales de...
Oh, it's not good. ¿Eso mejor se ve? Sí. Está... Ok, sí, más o menos funciona. Gracias.
Y ese complejo corresponde a lo que lo estamos tratando de ver aquí, digamos, ferromagnéticas, pero les tendré que tomar un poco de, si no biología, algo que tenga enlace, que haga ¿Cómo estás?
Vale, ahorita nos conectamos. ¿Usted también controla el volumen? A veces ha estado muy alto para varios speakers. Entonces ahí póngale atención. Si está muy alto, le digo que le bajo como Alfredo. Sobre todo en la tarde de ayer, había un cuarto que estaba muy ruidoso. Me apago ahorita y lo prendo. Yo lo deshabilito. Órale. A ver. Ya, a ver, probando. ¿El volumen está bien? ¿Así? ¿Está bueno este volumen o muy ruidoso? Ok, así le dejamos entonces. ¿Sí está bien? Ok, gracias. No, pues nada, lo que quieras, porque son en Harvard. ¿Qué tal? ¿Todo bien? Sí. Sale. Que me gradué de aquí, no sé, no de Harvard, gradué de aquí, doctorado en Estados Unidos, no sé, puede ser lo que quiera. ¿Qué tal? Órale, ¿y ahora estás aquí? Sí, me escribió, lo que pasa es que yo estuve, le tengo que contestar, decirle, oye, estoy en México, vamos a tomarnos un selfie y se lo mandamos.
Él está, ¿cómo se llama? Él ahora mismo está en Baltimore. Pasó con mi casa el otro día.
¿Sí?
Sí, pasó con mi casa, estuvo aquí, entonces me voy a para allá, para Baltimore. Iba a estar en un congreso en
Está en Filadelfia, por eso, mira, me acabo de escribir, a ver dónde está. Luis Montero, mira. Entonces le voy a decir. Así le voy a decir. Perdón, Luis, te perdí. ¿Cómo te llamas? Ok, perdón, te perdí porque estoy en México, pero me encontré. ¿Hacer sí? ¿Así? ¿Ese? ¿Así? Ok, perfecto. Pues ya, a ver qué contesta. No, pues mucho gusto, ¿eh? ¿Y qué eres? ¿Entonces eres teórica? ¿Tú también? Órale. Ahorita cotorreamos. ¿Quieres probar la tuya, papá?
Sí, recién la probé, pero me pareció que se veía muy mal, pero parece que porque estaba muy cerca. Porque yo la miré desde aquí y no desde allí.
A ver,
Pásamela. No pasa nada, sácalo. Sí, yo lo había sacado recién ahí. Dime, baja ahí, dime si se ve bien o no. Por eso no. Ah, está muy jodido de arriba y abajo, ¿no?
lo tienes en... No entiendo por qué. Pásame tu presentación.
No, aquí tengo demasiadas cosas. Espera.
Está raro, ¿eh?
Sí, no, pero recién me hacía otra cosa distinta que esto. A ver. Ahora lo voy a agarrar. Ahí está. ¿Ves? Y está corrido, ¿no? Por arriba. Tiene la resolución todo mal, ¿no?
Mira, pues dale displays, maestro.
Sí, es lo que estoy haciendo. Ahí está. Claro, ven, mira cómo está.
A ver, ¿quieres ver cuál le tiene la mía?
Espera porque ni
¿Por
siquiera
qué no le
sé.
pones duplicate displays, papá?
Sí. ¿Dónde está?
Display
El
arrangement,
otro.
ponle arrangement, ponle mirror. ¿Ok? Eso,
Claro, mira lo que está haciendo. ¿Ves?
Y yo creo que el problema es este 75. Ahí.
¿cuál? Ponle 800 por 600.
El Hertz.
Sí, exactamente.
Ahí.
Ahí.
¿Y cuánto necesitas?
60.
A ver, dale. Ok. Ojalá. De buen güey. Ahí estás, güero.
Sí, pero mira la resolución.
Ah, esa es la que yo tengo. Esa sí ya está.
Sí,
Ahora,
¿te parece que está bien?
Proyecta.
Así está en tu...
Vamos a ver. Proyectalo y te digo cuál tengo yo también.
Ahí viene. Quizás.
Dale.
No, mira.
Dale.
Ah, está en la revés.
Sí, sí, pero igual... Qué mierda. Sí, espera. Ahí.
En la alineación.
Y ahora no hay, ahora ni siquiera
Dile best for display.
aquí, pero están las dos iguales, ¿no?
Que
O sea,
no la haga mirror, que no las haga.
entonces, que no haga mi error, ok. No, ahí está.
Ahora, a ver, proyecta.
Espera que vuelva, espera que... Ahí está. Arrangements.
Ahora sí ya está.
Ahora está un poco mejor, sí. Pero ese está en 75, que eso no quiero en 75, ese es el que quiero en 60. ¿No?
Sí.
O quizás no, porque ahí, no, porque el ancho está bien. Es la resolución que es el problema.
A ver, tira el tuyo otra vez. A lo mejor ahí.
Default for display, quizás. Sí, ahí está.
Default for display es eso, papá.
Ápale.
Ahora sí, dale.
Ahora sí.
Y te va, ahora sí pones swap displays.
¿Y cómo está
Pícale
el,
aquí, swap displays.
¿dónde está el,
Aquí. Pícale aquí, swap displays. Ahí.
¿Dónde estaba?
Ahí. Vuelve a poner el display. No, vuelve
Ah,
a
ok, lo pongo ahí, está, listo.
Ok. Ahora muévele ahí arriba.
Ahí está,
Swap display.
listo.
Y ahora sí ya quedó.
Es una porquería, pero bueno, ok, es lo que hay. Está mal, ¿no? No, ¿se ve malo? Se debería, mira, se debería ver, o sea, a ver, mira.
¿En serio que no quieres compartirlo la mía? ¿Tienes animaciones o qué?
Ah, bueno, eso está bien, listo. Perfecto. Tengo
Ok.
películas, hombre, yo soy de Hollywood.
Yo también tengo un par de películas, pero tienes razón. Bueno. Espérate, ya se jodió esto también. Se cayó el PowerPoint. Mira. Ahí está. Creo que es la misma resolución, güey.
Bueno, buenas tardes. Estamos en la última sesión. Días, perdón, todavía. Buenos días. Tenemos en la última sesión dos expositores. El primero es el profesor Alan, que no hay mucho que presentar porque es súper conocido por todos nosotros. Estudió aquí la licenciatura, estuvo en la Universidad de California, en Berkeley, haciendo el doctorado y ahora es profesor de Harvard. Lo que quisiera decir es, no sé si Alan es consciente de que durante su paso por la licenciatura, la gente que estuvo junto a él, de su misma generación y trabajando bajo de él, pues tuvo un impacto, ¿no? Y él ha servido como ejemplo para varias generaciones, lo cual agradecemos y agradecemos que cada vez que Carlos Amador hace el requerimiento, que venga y platique con nosotros y que nos presente sus últimos avances e investigaciones. Muchas gracias, les dejo con el profesor Alan.
Gracias. Gracias. ¿Me oyen bien? ¿Me oyen bien? Ok. Bueno, otra vez, como siempre, es un placer estar aquí. Creo que tenemos que decir algo de la facultad. Estoy completamente de acuerdo con Juan de Pablo. Este cuarto siempre me pone nervioso a mí también, exámenes y presentaciones y todo. Okay, so now I'm going to switch to English, which is the tradition of this workshop. And I'll tell you about our analysis of metabolism. The title also talks about origins of life, but I could not cram origins of life and metabolism in a single talk. So next time I come, if Carlos invites me, I'll tell you about our work on origins of life. But I couldn't resist the fact that Alfredo was talking about snorkeling, and I actually have seen Alfredo snorkeling. So he and I were, this thing's not clicking. He was working. There's some software update here. Microsoft Auto Update. What? Sorry, PowerPoint. And actually, I happened to snorkel with Alfredo in Ecuador, so I want to show you a picture of him. Actually, he knows how to snorkel pretty well. And anyway, I will start with the overview of my research group in my research in chemical space. I like to talk about that when I'm here. Just to remind you what I think a lot about, and I think a lot about the immensity of space. There was a new planet just discovered yesterday in the closest star, which is pretty cool. With water, apparently. And if you want to think about, I already told you this information, so somebody should know how many how many atoms are in the visible universe. Does anybody remember from my last lecture here? Well, I'm going to ask you, Cersei, how many atoms are in the visible universe? 10 to the how many? Just guess. 10 to the 21? There's 10 to the 82 atoms in the solar universe. It's actually a number that we want to remember, because if you want to think about how many molecules can we make or study, or the size of synthetically available chemical space, any guess again, what's the size of possibly synthesizable molecules in about 10 organic steps? Well, there you could probably have gotten a good guess, because it's between 10 to the 6 and 10 to the 180 according to many different estimates. So roughly the same amount of atoms in the universe that we can see is the same amount of molecules that we can make. So organic chemistry colleagues are never going to lose their jobs. So that's actually pretty good. And in my group, what we think about is how to map this thing. And it's so huge, it's so incredibly large space. So how can I actually navigate it in terms of functionality? It's one of the big things that we do. Also, we're thinking more and more about synthesis and mechanisms. How to connect molecules to each other in terms of how close they are in this humongous potential energy surface. In principle, you can think about the entire chemical space as an infinite potential energy surface, where atoms are coming in around and reacting. Okay? And finally, some of the stuff I'll tell you today, it's just a glimpse because it's really not the topic, is how can we automate quantum chemistry prediction. So I want to get all these quantum chemists that the only thing they do is give you a DFT method and a basis set and all that out of business. That should be automated by a computer. So if you give you a molecule, the computer should be able to tell you what's the best basis set, dispersion method, etc. Not a human that is actually suboptimal for making those choices. So this is an overview of the things I've talked about here before. We like to think about chemical space in terms of functionality for solar cells, for batteries, for light-emitting diodes, and also for the origins of life and metabolism. So today I'll tell you about metabolism, and the main reason is because this is research done by two Mexicans in my lab, Benjamin Sanchez Lengeling and Adrian Kinich. So I wanted to highlight today the research of two of my Mexican graduate students. So this is the map of metabolism. Okay, it's amazing. So chemical space is huge, but metabolism is also amazing. You can see all these different reactions there. Okay, for example, you can see these major cycles that are very famous, like the Krebs cycle and so on, that are basically energetic. My view of life is that life started as a very interesting energetic cascade, and then it started building information. So people believe it's the opposite, but that doesn't make any sense, because you have to have energy before you have to store the information. And this is an example. What you saw zoomed there was one of the reactions that we're going to look at. So it's like this humongous subway map of reactions that actually makes living systems. And it's surprisingly little what we know about it. Okay? So how do we know about metabolic engineering? Well, the first thing we want to think about is, the first thing that we're going to do is hack it, right? I like a lot engineering nowadays. And the same way I like to hack molecules, I would like to hack metabolism. For example,
these particular pathways proposed by Barry Even and many other people for basically fixating CO2 are not thermodynamically feasible under physiologically relevant conditions. Okay? But what they did in these papers is they said, well, if they are not thermodynamically relevant, we can just take metabolism and hack away some pathways and now make it thermodynamically feasible. So this is kind of like the long-term goal of what I want to do here. I want to have the information so I can help the biologists in hacking the metabolic system for useful ways, for useful means. So where does it start? It starts with the work of Krebs and Kornberg. A beautiful story is that actually my graduate student Adrian actually tracked Professor Kornberg. He's actually a retired professor in Boston, and he has these selfies I don't have for this talk, but he talked to him and he talked about his research, Kornberg with Krebs. And in this paper also by Barry Even and Milot and others, you can actually see the view that I really like about this, because I've been thinking a lot about batteries. I think about life as this cascade of redox processes, okay? So you start oxidizing and reducing molecules in these different redox ranges, okay? And this talk is going to be about that. It's going to be about, okay, how can we think about the molecules being oxidized and reduced in this complex metabolic system, okay? In particular, redox reactions are at the heart of cellular metabolism, okay? In principle, again, life can be thought about as we're like these big batteries and we're actually just separating charges and actually using redox processes to actually be alive, okay? And not surprisingly then, 40% of the known metabolic reactions are redox reactions. Okay? So this is the enzyme catalyzed set of reactions. There are reactions that are not enzyme catalyzed, there are reactions that are enzyme catalyzed. And enzymologists have catalyzed it into different categories. Okay? Categories, for example, ligases, oxidoreductases, which is the one that we'll be talking about a lot, isomerases, lyases, etc. So the research of Adrian and I has been, can we map this entire map? Okay, we want to make the entire subwave map available with quantum chemistry, all the thermodynamics of it. That's the main goal. But today I'll tell you just about the oxidoreductase family, okay, all the reactions that are actually, and also intramolecular oxidoreductases as well. I guess that's what we zoomed in there. But basically reactions that happen to do with redox in these branches. Okay? So how much do we know? Okay? So here's again a zoom of this beautiful plot that really inspired us in this paper. Okay, here you see the different pathways. I apologize if the resolution is not as clear, but here is carbonyl reduction, NADP reduction, carbonyl to hydrocarbon, etc. We'll go into them. I'm going to be calling these different reactions different groups. This is group 1. Okay, we go from this acid to this ketone. We go from the ketone to the alcohol. That's going to be group 2 and group 3. And group 4 is going to be alcohol to hydrocarbon. Okay, so these are the different processes that happen, as you see, at different places in the redox scale. Okay, and the way we think about it in terms of our common chemistry is that we, of course, cook with olive oil and drink wine and drink beer and drink pulque and so on. And basically it's not surprising because these are all biochemical processes that actually are done by organisms around us. So when you actually think about vinegar to ketones, and then ketones to tequila and tequila to gas, all these transformations are done in biological systems. Okay, this is kind of a simplification, but it gives you an idea of where in the energetic scale of the metabolism these type of transformations can happen. Okay, so the challenge of quantum chemistry is that I need to do calculations of the free energies of processes that look like this. For example, the reaction of glycerate to hydroxypyruvate, mediated by NAD and NADH, will require QMMM type calculations of these huge beasts in water and good error cancellation to be able to actually calculate this free energy, standard free energy of reaction. Okay? So what we do is, of course, we calculate half reactions. And for example, we can spend a lot of computer time calculating this one, or we can actually parameterize it if necessary. But then we want to concentrate on the half reactions that involve the small molecules and not necessarily the big NADs and so on. So the first thing you do is you go back to your general chemistry that I learned here when I was an undergrad and partition your full redox reaction into 2 half reactions and then calculate the E-zeros of these 2 half reactions, which is just breaking the thermodynamics in these 2 pieces. Okay? So just to remind you, we have the Gibbs reaction free energy. It is related to the equilibrium constant, okay? And not surprisingly, this is how these things are measured. We're going to come back to it. In the 60s and 70s, and when people actually didn't care too much about nature papers and they were actually just trying to learn about nature, what they were doing, they were actually mixing these things with the enzymes and calculating the equilibrium constants. And so these things are very old experiments where from here they backtrack the free energy. And Alberti from MIT, former Alberti from MIT, spent a lot of time looking also at the pH transformations to actually get the right standard redox free energies. And of course, you remember that to go from free energy to E0, you need to figure out how many electrons there are. This is one of the things that my undergrads at Harvard don't get right. And you guys also probably don't get right if you study this. If you forget about this in the number of electrons, you get your exams wrong. Okay? And then this is basically how you come up with the standard Gibbs reaction. So again, thermodynamics from like second grade here. Okay? So, well, of course, I'm a theoretician, so I'll tell you about this because experiment is incomplete. So how many reactions there are? There is about 6,000 standard metabolic reactions. Of course, there's many more esoteric ones, but for a generic organism, there's about 6,000, say, E. coli. Okay? And then the NIST database contains equilibrium constants, about 400 of them. And then Alberti, which was doing research on this, he has formation energies, which again, from the formation energies, we can backtrack the reactions of about 200 compounds. This is about 10% experimental coverage. So it is amazing that the system, two things that are amazing to me. One, that we the quantum chemists have focused on the enzymes. We already saw many talks about enzymes here. And it's amazing. I mean, the work that we heard about enzymes here is just amazing. And the fact that we have so much power to actually study enzymes, like for example in Andrea's talk, right? But so little we have done just on the metabolites. That's where I saw the opportunity because I'm very stupid at doing enzymes. So I said, I might as well do the metabolites that nobody's paying attention to. Okay? And the second thing is how little we have done about that, and also, and so that was a big opportunity. And I forget what was the second thing I was going to say, but anyway. So again, here is the NIST database. Here is the Alberti's database. I guess I repeated. As a matter of fact, this is the cleaner version of that slide that I was doing in the plane, sorry. So that's about 10% experimental coverage. So what do biochemists do, or also like, kind of cheminformatics type people? They will say, well, why don't I fit this as group contributions? Okay, if it has a carbonyl, or if it has a sulfonate, or if it has this and that, it's going to add certain amount of energy to the standard Gibbs energy. So basically this is an addition over different groups, and you have a delta G0 for each group. Okay, and then you have this linear regression to experimental data, right? So that's what people do in the in the metabolic reactions. It's not surprising that you keep seeing this guy. He's going to be the postdoc advisor of Adrian, and Adrian spends a summer with Ron Milot, and he was the one that convinced me to get into all of this. So if you're a graduate student, I guess Adrian Roitberg just told me yesterday a story. If you're a grad student and you have a good idea, go and pester your advisor, and sometimes your advisor is going to tell you, yes, let's do it. So Adrian Kinich was a systems biology student, wanted to work with me. And I said, dude, and I actually met him at the Manu Chao concert, actually. He's like, I want to be your student. I was like, but you're a systems biologist, I'm a chemist, and this is what came out of it. So don't be afraid to do those kind of things. Okay? So, so you have different ranges of pH. Arren, that guy, is going to try to automate with robots the measurement of these metabolic reactions. And he also was the one that started thinking about group contribution. Errors of 2 to the 3 kcal per mole, that's amazing, okay, for actually such a shitty method, okay, to actually be able to get 2 to the 3 kcals per mole, okay. So what is our objective? To use quantum chemistry, that's the objective of the PhD of Adrian and also part of the PhD of Benjamin, to discover the principles in this biochemical redox thermodynamics. So you can pick your poison, you can either optimize geometries of these complexes, this is an example of an equilibrium geometry, or you can actually do MD and then look at the dynamics of the process and do free energy perturbation theory or other methods. We happen to be working on both. This is unpublished still. This is also unpublished actually, most of it, but I'm going to tell you just about this piece also for time. But, so anyway, how did you do it with equilibrium geometries? Well, for example, you have your reaction. This is fructose 1,6-biphosphate to glyceraldehyde 3-phosphate and the hydroacetone phosphate. I've been learning a lot about these names from Adrian. You have to think about all the pseudo isomers with the different charges, the conformers. Okay, you optimize all of them and do normal mode analysis. Of course, this Not perfect because there are soft modes that you don't discover very well with harmonics, but it doesn't matter. You try to do it. You do the thermodynamics of it, and you end up getting a delta G0 from ab initio methods. So many people have tried to do this, and we are the ones that said, okay, we're the first ones to actually go ahead and look at 3 different groups: enzyme class 2, enzyme class 4, enzyme class 5, 47, 8, and 58 methods with quantum chemistry, and we sent it to nice journals, and then we ended up in scientific reports. Why is that? Because look at our error bars. In the first approach at this, we had very high error bars. This is just quantum chemistry, unadulterated quantum chemistry. Okay? Huge error bars in the free energy compared to what we want, 5 to 10 kcal per mole. So we're going to try to dissect the errors again. Okay? We noticed, for example, that the charges were a big issue. Notice how, as there are more and more phosphates and charged reactions, you get more and more deviations in this box plot. So charge is a big issue, of course, when you do this kind of QMMF type of analysis. Okay? But what was really nice is we developed all this software to do this in high throughput. Okay? So now the question was, what's the best method? I mean, that was the first method. Now the idea was to publish this paper and get all the quantum chemists excited and get the Truras of the world and stuff, trying to get this better and better. Okay? That was our idea. But now, at the meantime, I've been talking to you guys about my work on batteries, and this is where things got interesting. First thing is we met and tell Adrian, let's think about first about the redox reactions, let's just forget about everything else, and then just look at them as half reactions. The same way we were successful at discovering our batteries. I told you about here before, if you come to my previous talks, about the fact that we screened about 10,000 quinone molecules to make the world's cheapest battery, which is an organic battery. It was published in 2014 around the same time. We synthesized and made the molecule, and then by collaborating with experimentalists, every week meeting with them, we discovered this molecule that I like to call anthraquinone disulfonate, or Jesus Christ quinone. Again, this is a generation 1 of our technology, and now it's a huge field of these organic flow batteries. Because I still claim these are the cheapest batteries in the world, not metals, but these type of molecules. Just to advertise, how do we do it? Well, we go back to a paper of Dewar. Dewar was mentioned here before in this symposium a lot. Dewar had a paper in 1969 in tetrahedron where he just said, come on, just calculate the enthalpy and correlate it to the standard redox potential. Just do a linear regression. Okay? And we did the same. This is actually our regression for the quinones. We have 1 millivolt accuracy and this huge R square of 0.989. So I can predict the redox potential of a quinone with just a simple quantum chemistry calculation, even semi-empirical. Okay? And of course, this is trying to take you back into why we want to go into metabolism of redox reactions. We were working on other type of molecules, and we found, recently we published, actually 2 weeks ago in Nature Energy, a vitamin battery with aloxacin. Okay? So it's basically variants of vitamin B12, so now we use vitamins actually as good redox electrolytes. So think about it now, you are using batteries made of quinones that are in rhubarb and in pheromones. Now you're using vitamins. Guess what, guys? Let's look at the entire metabolism. We're going to find more batteries there made of all the other stuff that we eat and shit. Okay? So basically, then Adriana announced to say, okay, well, let's correct things. Okay? So we're going to do the same trick as Dewar did and just calculate the delta E. Okay? And just basically ignore all the other terms. Assume that the difference between the reduced and oxidized forms more or less cancels, that the entropy and all the different rotational, translational entropy, vibrational entropy is the same. Okay, so here are group 1, group 3, and group 4. Delta E is correlated to the delta G zeros for the experimental data that we have. Okay? And I think this was just the slope, sorry, just the intercept and not the slope. Not even, I mean, one of the 2 parameters. I'll just back to it. Because it should be even on top of the line more. Okay? So this is basically our correlation. So just basically by doing the same thing as Dewar did, we can do it. Now let's compare it with group contribution methods. Okay? Here is group contribution. You can tell why it's wrong. I mean, group contribution, it doesn't have a lot of resolution about molecular structure. Isomers, for example, come up like in all these different bands. That's group contribution for you. Okay? So mean absolute error of 45 millielectron volts in the redox potentials. So now we do the quantum chemistry without any fitting, and then you get this crappy thing. You fit, in this particular case, I just fit just the intercept, and I get this, and I fit the intercept and the slope, just a linear regression, then I get better fit. And that's what we do. We have 2 parameter fit for these reactions. Okay? But notice that there are some little outliers there. Anyway, okay, now let me just go back to something else. Now here comes Benjamin. Benjamin's PhD project is the project I told you about to get all the theoretical chemists out of business. Okay? So his PhD project is you will never have to pick a basis set and a functional again. Okay? The computer will pick it for you. So Benjamin has done for many different things, but the idea is to actually go ahead and try it for metabolism too. Are you going to do quantum Monte Carlo, DFT, hard trifactor, closest semi-empirical, B, half and half, or the functional of Alberto Vela? Which are you going to do? And this is actually the choices that we have when we're doing metabolism. Okay? So I think for the first paper, we actually chose the ones that are going to be boxed here. No, they're not boxed here yet. But we're going to do the single point energies with DPLNO or with MP2. We're going to use your favorite functional or the ones we employed, these basis sets, how many waters we're going to add. Are we going to have counter ions? Are we going to protonate things or not? What a mess. So that's the problem. Adrian spent so much time actually doing this by hand. And it's not clear that a human really knows what's the right answer. Okay? So I call it the 2 to the pick your poison problem. Every time you have a choice, you have an exponential growth in the number of choices. So every time any of you invents a functional, then you exponentially keep growing the choices for the quantum chemists. So don't do that. So I guess this is basically what we did for the first paper, I guess. Okay? So this is again the goal of Adrian. Okay? We want to have a system where you say, please give me the redox potential for the uploaded molecules in English, and then just does the quantum chemistry for you. So input files should be text. And we're getting there. We're going to get there one day. Okay? So there's never going to be an input file for your code anymore. There's going to be English. Siri, can you please give me the redox potential for this, please? And then you just go and hit Tax al Pastor. So the way Benjamin implemented all of this, he says, okay, let's go do ORCA. We like this code a lot. We automatically send our stuff to the NSF computers or to our own Harvard cluster, compare the thermodynamics again, and we go back to model chemistry space and explore and explore and explore. Turns out that in this case, Adrian invented a method that is called stochastic line search that was better than Bayesian optimization, which is what Adrian was doing. Sorry, Benjamin was doing. Benjamin now is improving and so on. So we're working on this. Well, the idea is, imagine you have this set of choices, and actually this is the one that we optimize over. And you go one particular dimension, the one you think is the most important, optimize what's the best method there, optimize what's the next best choice in the second one, in the third one, in the fourth one, in the fifth one, and so on. And that's how you come up with your approach. So this was the best thing we could do there. It beats more fancy methods, which is interesting, but that's how we picked the right thing to do, at least on the menu that we had that day. So we go ahead and do that, and we have now our best model chemistry, and we regress. And then see, Cambit relief, MO60, PSS, B3, blah, blah, blah. Look at these guys here. Okay? What's up with those guys? They do not resist the power of quantum chemistry. Okay? Okay, so what's going on? So we started doing statistics and say, well, those guys must be experimental errors. Or quantum chemistry cannot attack them, right? I mean, so it's just 2 choices. I cannot rule out that other choice. So we look at the z-value, which is a very simple way of renormalizing the errors. And we actually do then cheminformatics versus quantum chemistry. So cheminformatics is another way that we're working on predicting the redox potentials with another postdoc in my group. And we notice that when the z-values are correlated, large z-values positive or negative, that means that 2 completely different methods, and this can be an hypercube or all the methods that you try. You go to the corners of the hypercube, and if you see data points that lie there, they probably are experimental errors. So we remove those guys. Goodbye. Okay? And therefore, we remove these outliers, and for these G2 reactions, and I try to show you examples of the reactions of the different groups. For example, this one is important for carbon fixation. Now we take away these 5 or 6 or whatever data points that we believe are experiments from the 60s, from a lazy graduate student that didn't do it correctly. Okay? So anyway, that's it. And that's it. Basically, then our fits now are to these guys. I'm forgetting about these ones, and we get much better regression. So the mean absolute error only goes down to only 2 milli-electron volts, but the statistical predictive power is actually higher. So now, these are the optimal model chemistries that we decided to do with this stochastic line search algorithm. You can fight with me, but then if that's the case, throw it at the same method and show me why yours is better. I mean, it doesn't really matter after all. We do the fitting to the experiment anyway. But this is basically what we found was the most successful thing to do. So actually it's paradoxical that instead of adding explicit waters, it's better to use an implicit solvent here. We actually carry less error, okay, for example, okay, in this case. So we have created computationally the database of the non-measured metabolic redox reactions. So we have the best database for that. We have 6,000 non-measured redox reactions, 7,700 with less than 7 carbons, and this basically implies there is about 10,000 quantum chemistry calculations that you need to do to run this database. And of course, every time you change the method, you have to redo it. Okay, but now here is our own plot of Arendt's paper. Here are the different reaction classes. These are the 100 measured reactions. And here are the 700 not measured that we have in our database. Okay, now calculating the database. So these are our predictions. Okay, so you see this is the range of the measured ones. This is the range of the ones that we know are in the database, but nobody knows the number for it. Okay, and group contribution could give you some idea for this E0, but we have some more resolution. So now I'm going to spend some of the time in my talk showing you some of those things. Okay, so the first thing is that, for example, I have this quantum chemistry, and I'm going to go to the first group from ketones. I want to open up the ketones, okay, and in particular I'll go to the, for example, here we have the lactones, and here we have all these different keto acids that have 1, 2, 3, 4, 5, etc. carbon separations. So we believe for the first time we have enough resolution to actually give you the E0s for different subfamilies of redox reactions. Okay, so that's pretty useful for the metabolic engineers, because you might want to make a fuel, or you might want to make a material that has 1, 2, 3, or 4 carbons. You want to know what is exactly the right free energy that you want to just place your metabolic engineering pathways, so that you actually have enough energy to go and make this particular bioproduct or this particular bioproduct, etc. There are interesting ranges for the batteries around here, so you might be actually thinking about the type of molecules that could be the next battery. Okay, and don't scoop me. So here's another example. This particular set of reactions. Now here we have conversion of these carbonyls to amines, for example. So this is the type of reactions that we're looking in this particular group. So again, what Adrian has been doing a lot is now thinking about, well, if you have a phosphate, this is the range. If you have vicinal tricarbonyls, you're here. And then we further expand and we see the box plots for these 3 different groups, okay? And gives us an idea of what did nature evolve to having, in different places of the very, very accessible part of the water window, which is where we actually operate metabolically. Okay? So again, these are the different types of reactions and how they're cataloged. So now we're going to go to this one. Again, I'm just showing you data, especially if there's any people that are interested in metabolism. Here, the high potential, okay, is close. It's also very interesting because molecules with the high potential have to be more resistant to water. So anyway, from the battery perspective, it's quite interesting. These things start to look more and more like some of our battery materials as well. But here, for example, the hydroxy acids, okay, benzene derivatives, alcohols, etc. Okay? So again, this is just to try to give you an idea of where all these different things have been lying out in our database. So I guess I actually spoke quite fast. This is the first time I finished in time, I guess, here at UNAM, I don't know. When I summarize what we did so far, we have these new methods in my group that are still not published, and we're working on them. And sometimes what Benjamin is doing is much better, is Bayesian optimization. For example, for dispersion forces, it's actually pretty fun to do that. You take a database of dispersion interactions, and then you think, Grimme, are you really right? Or so-and-so is really right? Let's have the computer be completely unbiased, and a computer make all the choices and see if Grimme is right, or we are right, or with many-body dispersion, or who is right. By the way, many-body dispersion invented by a Mexican, Russian, Alexander Tkachenko, who should also come here one day too. Now, we also really like this. We're going to write these papers on this systematic removal of outliers. We believe there should be a tool online that you upload your stuff, and then we tell you what is a good outlier or not.
We believe quantum chemistry will become more and more important for metabolic analysis. I think the metabolic people still don't know what to make about quantum chemistry. They have not been, the system biologists have not been exposed to it so much from our side. Of course, perhaps from the cancer side and other sides, yes, but from our side it has been kind of like, what do you mean by quantum chemistry? What can it do, etc. Okay, and I think we were going to be able to, I guess I repeated this, but quantum chemistry will provide new insights for us at least into this particular set of redox reactions and hopefully in the rest of metabolism. So I want to show you, again, this is Adrian, which I've been mentioning a lot in my office with me. This is Benjamin, and this is myself. And we call it Team Mexico. There's a student of mine that is not pictured here, Teresa Tamayo, which is hails from Guadalajara, and she's one of my first year graduate students too. So right now we have 3 Mexicans in the lab. So I just want to remind you that if you guys want to go study abroad, you should do that. You should apply, and sometimes you're accepted if you have good grades and good everything. And we can help you. Every time somebody sends me an email, I immediately click forward, and he or he will reply to you. But now there's a problem here. Something here. What is that? So we live in the United States, and we actually found this Pokemon in my office. Okay. So then we found this Trump Pokemon. And that's what we did, okay? So that's how we think about this motherfucker, okay? And with that, I'm just going to show you my research group. Again, here's Benjamin, here's Adrian, people that have worked on this. Actually, I forget her name, visiting professor from Iran also worked on this project, etc. I don't think Tere is still in this photo, but so with that, I'm going to end, and I think I have some time for questions. Thank you.
Thank you for the great talk. I hope you don't take us out of business. Okay. I was just wondering, when you talk about these experimental things that they are outside the trend, for instance, I remember that once we had this experience with excitation energies that DDD-FT agreed with linear couplet cluster, and they were both wrong. And the only thing that gave us the right answer was multi-configurational perturbation theory. So how do you think, how do you...
I said that I don't know if quantum chemistry cannot attack it or if it's an experimental error. So I've been loosely calling them experimental errors, which could also be that my quantum chemistry cannot attack it and I need a more sophisticated method. Correctly so, of course. You don't know which one of the two it is. But obviously, if you're fitting your methods and you have that data point that is unruly, you probably don't want to use it. You don't believe that data for either one of the two reasons. Either your method is not predictive there, or you say the experimentalist made a mistake.
Thank you.
Of course, that's kind of, you cannot rule out the fact that, quantum chemistry cannot solve it.
Hi, Alan. You mentioned that as you increase the variables in the system, you exponentially grow. But I was wondering, how do you know that when you do this reaction, you have a global minimum and not a local one? Because as you go to these reactions and you increase the number of atoms and so on, you will have a lot of local minimum energy. How do you check that?
So as I mentioned, we calculate, basically what we do is a thermodynamic analysis. So we generate a bunch of conformers and we Boltzmann weight them. So there's many times that the molecule has many conformers that are active. My favorite example is a paper by Vincenzo Barone that says, why is eggplant purple? If you have ever heard him speak, it's amazing. So eggplant is purple because it's actually switching between two different conformers all the time, and both of them absorb at two different colors, right? But of course the color of the molecule is this overlap of the two absorption peaks. So he was figuring out why some people calculated eggplant and it was green or purple. So here we do the same. We calculate all the conformers,
We Boltzmann weight them, and at each conformer we do the thermodynamic analysis and we add up all the terms. So it's not one minimum, it's actually an ensemble of them. So yes, so that's how we do it. In the molecular dynamics method that I didn't tell you about, that my student Flo and Adriana and others are working on, then we do everything with tools like free energy perturbation theory or just getting spectral densities and integrating thermodynamics out of them. And there, of course, the molecule is floppy, and we start from different starting points, and hopefully the MD covers enough of the conformational space that we're safe.
You are doing thousands of reactions, and probably you have thousands of pathways, no? That's why it's, I don't know how many calculations you need to do to really go to the global minimum. That's
No, I mean, this is very small molecules. I mean, these are small molecules. The conformer generators are pretty good at generating good guesses for the equilibrium geometries. So we actually get about, say, order of 20 conformers, and then do the quantum chemistry on all of them. Then we take the lowest energy ones, and then Boltzmann weight them. I don't think we're making too much error on the conformers. I'm much more worried about the errors that we make on the charges and interaction with solvent and very large molecules like NADH, and then, you know. So we have errors that we worry more about, I think, than the conformers.
Well, you're keeping only the electronic part for the calculation of the effective, right? So you have to balance very well what you're doing in terms of reactions and products. Have you thought of using isothermal reactions, for example? In some cases, I know you're using a lot of molecules, a lot of reactions, but that probably would improve your correlations. What do you think about this?
Using isodesmic methods. I mean, there are some reactions that isomerases, for example, are kind of like isodesmic type. You know, we also have looked at that subclass, and that's the one that we have the least error in isomerases. And actually, group contribution actually is the worst there, so it's a good area. But yeah, we could potentially use other methods like isodesmic tool, isodesmic tricks. Maybe we should look into that. Yeah. I mean, right now, it is always a balance of what do you do and how fast can you cover chemical space. And here, this is where we are right now. But yeah, the reason that we open up these databases is that maybe other groups like yours can go in and find a better method, and then tell us, and then upload it to the database. And so there will be a database soon we'll be releasing, and then everybody can go and add more data or whatever.
Very nice talk, Alan. So do you always look at absolute errors? Because really chemistry in most cases, even in redox reactions, are about one quantity with respect to another. So even if you have large errors, but they're constant, then you're safe, right?
Well, in some sense, I mean, it's all relative, right? We're calculating the E0, which is the difference between two reactants and products, so we have error cancellation there. There is an absolute error that we actually achieve, because people never want to see an E0 wrong, right? I mean, they want to see, if you predict 300 milli-electron volts, you probably want to be at 300 milli-electron volts. So we're happy with those two parameters. A little bit of slope, you know, and then a very, very small slope change, and then just a fit. So I don't know if that answers your question, but they are really relative errors, because we're calculating difference between products and reactants. So there's a lot of error cancellation in the DFT there, right? Is that what you're asking? If not, then, yeah. No. Okay.
Hi, Professor. Thank you very much. So I'm just wondering, what are some applications of making these databases? Like, how is it just a theoretical study, or do you have in mind some applications of calculating these delta G values for many reactions?
I did mention already that I want to eventually collaborate with metabolic engineers, and then work with them and say, well, you really want to make this, fuel, then which pathways you want to knock out in your organism. Okay. And for that, then you want to know if you knock out the pathway or not, if the reaction is thermodynamically accessible or not. And then other people that know about enzymes, like Adrian or Andres, will help them figure out what's the best enzyme for the best rates. And I leave that work for them. And I just tell them the thermodynamics of the reagents is this, so you can do this. So they don't spend a lot of time doing something that is uphill, and maybe they will have low yields, right, just by the Chatelier's principle, right?
Thanks.
Alan.
You went too fast, but if I got the message correctly, the conclusion of the best methods that the machine selected were PV0, that was one, and the other was for the solvation, it was Cosmo, right?
That's correct.
Well, I just want to underline that that's really nice, and that tells you that we are not out of business because, well, no, not those ones, the previous one. Before you said there was one there. It's
Okay,
PV0
Def 2BP.
and for the solvation, Cosmo.
Yeah.
Regarding PV0, I just want to stress that it's and that's for the students, it's a non-empirical functional. It's not B3 leap. Fortunately, it's not B3 leap. So please don't use B3 leap. Use, and for a reference, I will, from now on, I will refer to Professor Aspuru as the reference. Please don't use B3 leap.
No, if my computer tells me, no, I disagree with you. If my computer tells me to use B3 leap, I will use it.
Oh,
so
shit.
your method,
No.
I'll add your method to my list.
We have to talk
and
about
if your
that.
PVE method is better than B3 leap for my application, I
That's
will use
fine.
it.
Okay,
I am a molecular engineer. I'm not the guy that fights about functionals. I love
right,
the work that you guys do, but I throw it on my computer, and my computer will tell me what's the best functional.
Great,
And
okay.
I don't want to get into this fight between you and Kieron, Joe and Kieron versus Axel Becky and whatever. That's not my business.
No, I'm not fighting against Kieron, but Axel I will. But anyway, implicit solvation. It's a little bit discouraging, right? Because the common knowledge nowadays is that actually a mixture of, and I don't know if this is the question, of explicit solvation and implicit solvation is actually the best way to describe a solvated molecule. Did you consider that possibility or only explicit and implicit?
That's a possibility, and this one actually, the method actually adds 4
Also,
waters.
so then it's
The method has 4 waters there. I mean, that's what was optimal. The issue is, if you have explicit waters and you have a very large anion, the anion actually has to be localized over the waters a little bit. It has to polarize correctly the waters and then the polarizable solvent. We believe there's a lot of error there. And also, how do you deal with counter ions? So there's all these questions that at least I have that some perhaps bias my model to actually go to, in this case, 4 waters and Cosmo.
Okay, so then your conclusion is that these mixed solvation models are the best. Explicit together with implicit.
It's just that I forget if this is one of the optimizations that we did and this is what came out. And in another optimization, we ended up with the Cosmo with 0 waters.
Okay.
So the errors are very close to each other, depending on what optimization you do. So I think the rest of the data I showed you is with this. Okay? So, and that was the output of another optimization. This is what happens when your grad students give you slides and you still don't know exactly which one is which because we haven't written the paper. But I think this is the one from most of the data that I showed you, and the other one is the result of one of the optimizations.
Well, finally, I certainly would like to exponentially complicate your life, providing you with more non-empirical.
Yes, sure. I mean, I was going to tell you after your talk
database.
that we should put it into QChem and we should put it into Orca and stuff. I mean,
No, but you
I'm
said
a
Orca,
user.
right?
Sorry?
Orca?
If you have it in Orca, that's great. All right. We can put it in Orca. I mean, we can run it. I'm very honored, and I'll use your functional in my searches. But if it loses, it loses. And if it wins, I'll cite you. Also, if it loses, I'll cite you. Yeah, exactly. You'll come to the Olympics. Yeah, this is a promise. We'll add you to our. Yeah.
Thank you. Maybe we can agree that B3Leap can be called the I'm feeling lucky thing in Benjamin's Google. And that's what
Yeah, I mean,
okay.
yeah.
My question,
I'm not against it or in favor of it. I'm agnostic to it. Yeah.
my question is this one. Having 700 redox potentials that were not known before, it's a lot of information. It's a lot of chemical information.
Yeah.
Have you thought about what
Publishing in some nature journal?
No. What we can learn about chemistry?
Yes, that's what Adrian is
Because of
doing.
those things.
Well, that's what Adrian is doing. How many carbons?
Exactly.
How many carbons
Exactly.
are there? The paper is going to be all about that dissection of all of that. Yeah, that's what we want to get into. Yeah.
Do you have anything else already about that? That's really interesting.
No, Carlos, I don't think so. I think we so far just kind of dissected the different subclasses. But I think for each one of the cases, you will have to go to the biology of it and see if this organism picked to make this product versus this other one, and what enzymes are there, and that's something that of course we should do. But yeah, I think this is great, Carlos. Yeah, I think that's a good question, but we just started on this. Okay,
Okay.
so I'm going to pass it to Adriana.
Agradecemos al profesor Alan.
¿Quieres que esté? Es el de Alfredo. Ajá. ¿Lo pagaste? ¿Quieres pago?
Ok, llegamos a
No, me lleva.
la última
Hola.
conferencia de nuestro
Sí,
simposio.
solo.
Y tengo un gusto en presentar al doctor Adrián Roybeck, al profesor. Él trabaja actualmente en la Universidad de Florida. Él es originario de Argentina y estudió en la Universidad de Buenos Aires. El doctorado lo realizó en la Universidad de Illinois en Chicago y él es uno de los jefes de desarrollo de AMBER y de la sección de química teórica de la ACS. Él vino hace 2 años a darnos un taller de AMBER. Varios de nuestros alumnos y profesores e investigadores estuvieron en él y ahora viene a presentarnos su investigación. Muchas gracias.
¿Me escuchan ahí? ¿Es that okay? Good. Bueno, primero en español, como decimos. Muchas gracias a todos. Bueno, español, argentino hablo, que es otra cosa. Un dialecto extraño. Muchísimas gracias por la invitación a todos. Realmente es un placer estar aquí. So I'm going to switch a little bit about the talk that I'm going to give. So I was thinking a little bit about, people have stepped in here and gave final exams or something. I don't have that, okay? But it turns out there is a connection that is dear to my heart with Mexico and with Vietnam that most of you do not know about. So exactly 50 years ago, it was 1 month after one of the many coup d'etats in Argentina, there was something called La Noche de los Bastones Largos, the night of the big sticks, okay? And where the police basically took over the universities and the military. And the people that should have trained me when I was in school were all exiled, right? They resigned, they really kicked their butts. And it's appropriate that it's 50 years. And it turns out that many of them ended up in Mexico, and some of them even in UNAM. And Mexico actually historically has received a tremendous number of Argentinian exiles, right? Over all history. So, thank you. I know you're not personally responsible, but thank you. So let me tell you, so I'm going to give you a very short story of computational chemistry, right? This is Auguste Comte in 1830, and you can read that. Basically says, any attempt to employ mathematical methods in the study of chemical questions must be considered profoundly irrational and contrary to the spirit of chemistry. If mathematical analysis should ever hold a prominent place in chemistry, an aberration which is happily almost impossible, it would occasion a rapid and widespread degeneration of that science. So we're screwed, right? So the good news is that this guy, he was actually an astronomer. That's the good news. But he was a philosopher. And you could read his books, and there's some interesting things in there. But it turns out there is, again, a little connection to Latin America. This is the guy who wrote about order and progress, right? That was his big philosophical thing. And we know how this thing has been applied in politics in Latin America, right? Order and progress are very, very loaded words, right? Around, like, in order to have progress, you need to have order. This is dangerous. It turns out that 75 years ago, exactly, Conrad Zuse actually created the first automated machine, computer, right? They actually stopped building it. He built only one. It's Turing complete, fully programmable. And it turns out that the guy who proved that this is a universal Turing machine is Raúl Rojas González, which is he's a Mexican scientist, very famous, I understand. So again, another trick of history, right? So we go then to Iraq, right? And we all know this quote. He basically says, this is 1929. I'm going to talk about this paper in a second. 1929, he basically says, look, we're done, right? We know everything there is to know. The underlying physical laws necessary for the mathematical theory of a large part of physics and the whole of chemistry.
That's an interesting juxtaposition, right? Are completely known. And the problem is that, the exact application, there are equations that are too complicated to be solved. All right. The second part is actually even more interesting. It says it becomes desirable to design approximate methods to apply quantum mechanics, right? And then you can look at complex atomic systems without much computation. And that's actually, those two things should go together. What he meant by the first is that we finally know that it's all about Coulomb interactions. Right? The quantum chemistry is 1 over r, and we're done. And we don't know what to do with that information. We're learning, but mathematically we're okay. And then Henry Heiden put it in a different way, right? It says in 1944 that chemical questions are just applied mathematics. And he was wrong, but okay. So it turns out, I actually spent a little time a couple of days ago looking at that issue of the proceedings of the Royal Society. Look at the other stuff you can find in there. Hartree, right? The structure of the atomic nuclei with Gamow, with Rutherford. Crystal structures with Fourier series, Bragg. Molecular dimensions of organic compounds, Nassini, right? And then you can talk about total reflections at the interface between two media. So that issue is insane. It's absolutely, it's one issue of the Royal Society that is just absolutely crazy. So let me go back to what Dirac said and the way I've been thinking about it for a while. He basically says, look, we need to do approximate methods. But in my head, that actually means that we're wrong. We're always wrong. We're always going to be wrong, right? And so the trick is to be as exact as possible, but to essentially look at how do you manage approximations. Yes? Something that is dear to my heart. If you're going to be wrong, do it faster, right? Don't wait 5 years to find out you're wrong, right? So in the AMBER community, one of the things that in molecular dynamics, one of the things that I do is make methods really, really fast. And the way I describe it is that I provide a rope and a knife, and the user can do whatever they want with it. They can kill themselves or not, right? That's not my job. And then the question is, how wrong might you be, right? So thinking about accuracy and precision bounds is if you're going to be off by 10%, maybe you can live with it. But you're going to be systematically off by 500%, well, maybe you should do something else. George Box actually has this famous thing, right? Essentially, all models are wrong. Some are useful, right? And the question is which ones. One thing that I do a lot, and you can read that, I'm not going to read it, is Feynman's famous thing where he says, look, it's all about atoms jiggling and wiggling, right? It's all about things moving. Supposedly Galileo, when he was about to be killed and then he was pardoned, he said, I pur si muove. He was talking about Earth, right, or the heavens. But the idea is that most of what I do in science has to do with things that move, right? And it's actually a very strong reaction that I have, and I don't know where it comes from. I don't like 0 Kelvin things that don't move. Right? I just don't. So a lot of what drives me is trying to think about those kind of things. So let me tell you about science. I'm going to tell you about some excited state modeling of conjugated polymers and some things about classical MD. They're completely different things, except that they're both about movement. So this is the slide that you write when you send it to a program officer that's going to give you money, right? You promise them you're going to solve everything. But the idea is that, you get these nice cells, right, with cathodes and anodes, donors, acceptors, a little device. And the idea is that, in principle, we can create these antennas, these things that will absorb light, in principle match to the solar spectrum, and funnel that energy to the appropriate place in the device. So if you think about it, you will have many, many chromophores, and then the energy will just go down. The couple of things that you can do is to think about large absorption, many chromophores. The absorption has to be very broad to match the solar thing. You need to minimize non-radiated path. You don't want to absorb photons and then basically, having them up as heat. That will be a waste. You need to some kind of a bias, so the energy goes to where it wants to go, and this needs to be photostable. Right. So it turns out that in this business, there's two wide models. Sorry about that. And people actually, Alana and I have witnessed people fighting, literally screaming at each other over these things, okay? And I don't think it's that important. But, so one of them is that, you absorb a photon, you get a localized exciton, and this exciton basically goes downhill in energy, right? Being localized, and it dumps the energy somewhere. The other thing is that you get an exciton, and this exciton very immediately localizes very, very broadly, and then eventually focuses somewhere. So if you read these papers about quantum biology and why photosynthetic reaction centers use quantum, this is basically the underlying issue. So I'm going to show you something that we, from a programming point of view, that we wanted to solve a particular problem, the kind of things that we did. So in order to study these systems, you need the following. You need a system at room temperature. So it's moving. There is a set of conformations. You're going to absorb a photon at some wavelength. So you're going to end up in some excited states. And if you think about this, once you're in an excited state, let's say that S2 excited electronic states, if you just stay within the Bohr-Oppenheimer approximation, you're going to stay there forever. There's absolutely no way you're ever going to come down, right? Those states are exactly orthogonal. There is no coupling. You're dead. The only place the coupling comes from is called non-adiabatic. Coupling is because those things are moving to that intersection, and then at some point, they literally get confused, and they need to figure out, do I stay up or do I come down, right? That's the way these things relax, and eventually they actually fluoresce or they deposit the energy somewhere else. So in order to treat these kind of things, you need to do ground state molecular dynamics for sampling, you need to do electronic excitation, you need to do molecular dynamics in the excited states, and you need to include this process, right? And we want to do this for pretty big systems, about 1,000 atoms. We want to do this in the experimental timescales, which are ordered, they're fast, that's the good news, tens of picoseconds. And we want to do this efficiently, and we need to run thousands of trajectories for this thing to be really good. So you can read that. That's just technical issues, all the stuff that we had to put into this program. The most important thing here for the people that are experts in this is that the cost you pay is that you need to go down in accuracy, right? There is no way you can do all this with DFT with a big function on a huge basis set. Because if you do that, you're going to do 2 atoms. That's about it, okay? Which is very interesting. It's not where I wanted to go. So we spent a tremendous amount of time looking at semi-empiricals, reparameterizing semi-empiricals, and again, my original field is more about modeling in biology. You basically decide in a model-wise problem is where can you stay that gives you the correct physics, right? Compared to experiment, and then you keep analyzing. You don't have to go all the way out in methods. So right now we can do this kind of thing. So just to give you an idea, 10 picoseconds of excited state dynamics, 500 trajectories, a small time step for the electrons, you end up having 10 to the 9, 1 billion in American units, excited state calculations, right? For this, so you better pick some method that can actually take it. And we spent a lot of time coding and making this efficient. These are the systems that we're interested in. If you look at them chemically, the important thing is that there's a lot of symmetry in these things, which is pretty cool. And if you look at them carefully, I'm going to show that little segment. We're going to call it a 4 units. So it's a ring, CC triple bond, another ring, CC triple bond, and 2 more rings, right? So there's 4 rings in a row with the CC triple bond. Then they're meta-conjugated to a 3-ring unit, meta-conjugated to a 2-ring unit, right? And what happens with these architectures is that the excitations are extremely localized because the meta-conjugation breaks the overall conjugation. There is no wave function in this thing that is fully delocalized. They're all localized in little chunks, right? And we're going to play with that. And then I'm going to show you something about these other things where you have 4-ring units, and then you have something that is ortho to the previous one. So now there's conjugation all the way across, right? It's a different way of thinking about this. Experimentally, let me see if I have this. So experimentally, what happens with this system that is called the nanostar is that you absorb energy at about 320 nanometers. That's where all the two ring units actually absorb. And the fluorescence comes from the perylene in about 10 picoseconds with 0.99 quantum yield. Right? So this is extremely fast transfer across reasonably long distances with no loss of energy anywhere else. You don't lose energy to the solvent, you don't lose energy to anybody else. Right? And this thing actually emits at about 500 nanometers drop. So you've gone from photons at 320, emitting at 520. There's no overlap. The molecule has to do something in the middle. So what we have done so far, I'm going to show you, we have a bigger system, but I'm going to show you some things that are little pieces of this thing. 2, 3, 2, 3, 4, 2, 2, 3, 4. So we're studying little chunks of these to try to build what we understand. And then that weird one that I'm going to get to soon. You can compute the absorption spectra. Basically what we do is we run many molecular dynamics and look at the excitation energies. At low temperature, for this system, you get very defined peaks. They correspond exactly to the individual units. I'm going to show you that in a second. But what's interesting is that at 300 Kelvin, now these peaks overlap because these things start moving around. All right? And just to get you an idea, let's see. Well, actually, everybody can try to guess. That's fine. What's the rotational barrier for that, for a benzene ring in the system? It's very small. It's almost exactly kT. It's 0.59 kcal per mole, right? That's not a double bond. It's a triple bond. This is a cylinder. This thing can just rotate all the way around. So as soon as you include disorder, temperature, whatever you want to call it, these things actually change how they absorb, right? These are not flat by any possible means. And that's interesting because if you go and do an experiment and you, let's say, excite at 380 nanometers, you're not exciting a single state. You're actually exciting some combination of more than one, right? While at low temperature, these things are very, very well defined. So let me show you the localization of these excitations, since I try to convince you. Here we go. So these excitations that you're seeing here are the excitations from the ground state to the S1 state. That's localized in this 4-ring unit. The S2 state is localized in the 3-ring unit, and the other excited states are localized in the two state units. These things, when I say localized, I say mostly localized. They cannot be fully localized because if they were, there would be no coupling and there would be no transfer, right? So one of the things that happens, this is one trajectory, right? You excite in an excited state that is localized in the 2-ring unit, and as time goes by, boom, the energy just goes downhill. Right? And this is completely driven by movement. Those atoms, if I repeat this is a short trajectory, they're actually moving. And that is actually what's making the whole thing couple. If you look at the little hydrogens or whatever, you obviously see them, they're moving. So what can you do? You can take that system, the 2-3, you can run 500 trajectories, and you can look at the population in the 2-ring unit versus time, comes down, the population in the 3-ring unit goes up, as you go from the S2 to the S1 state. So what you are doing is, you excite an electronic state, you go down in energy, right, by coupling, and you go forward in space. So you transfer, you get a gradient to go down, and that essentially, because the way these things are built, means that the energy went from one chunk of space to another chunk of space. If you have these 2, 3, 4, kind of the same thing happens. If you look at the 4-ring unit, it starts with nothing in the beginning and it goes up. And the 3-ring unit goes up and a little bit down, right? So what happens is that you get this unidirectional energy transfer. You start in the S3 state, you localize in the 2-ring unit. At some point, the dynamics is such that the S3 and the S2 states are essentially degenerate. So now you delocalize the wave function. And sometimes you localize back at the top, and sometimes you actually transfer. And something that is really, really cute about this system is that we've shown it in many, many ways, is that once you transfer, once you go from the initial state, delocalize, to that intermediate state, the gap to go back increases by a tremendous amount. So you never go back. Right? And that basically means that this thing is completely unidirectional. I'm going to skip these for a second. This is another, nice little movie. for those trajectories. It's a little bit more complicated system where you transfer, again, down in energy, forward in space until you localize all the way down there. And what's interesting about this is the following. The way that most people would look at this data is the following. This is the population of the excited states, different excited states, versus time. And S1, which is localized in the following unit, starts with no population, and it goes up, so you get transfer into it, right? All the other states essentially start looking the same. They either go straight down, or they go a little bit up and a little bit down, right? And this is classic. I actually use this example in my physical chemistry classes. This is typical first-order kinetics, right, or sequential kinetics. You go into a state, you dump. They all look the same. So it's very boring. Essentially, as the molecules become large enough, they all look the same, and it's really hard to read. So we change it into a description in which we stop looking at the excited states and we simply ask in a spatial way, how much energy is in the two, how much of the excitation is in the 2, in the 3, and at the 4, and I don't care what the electronic state is. I want to know spatially where it is, right? And when you do that, things become much clearer. The Forling unit, it turns out that it has a little bit of density and it goes up. The 2 goes down, right? And then the one in the middle goes a little bit up and a little bit down. And if you look at it in detail, now you can start dissecting these things. You can say, look, how does the energy go from the beginning to the end? I can imagine 2 ways. I can go through the tritium units to the 4, or I can go directly, right? Well, we have all this data. We can look. We can dissect everyone and ask which path did you take. And it turns out that in this case, the answer is exactly 50-50, all right? And when people did experiments on this, I'm going to tell you what's in a second, they found that if they tried to fit their data, their experimental data, this is transient absorption, with just 2 rate constants, meaning 2 sequential transfers, they couldn't fit it. They needed something that they interpreted as a direct transfer, and it agrees exactly with our experiments. The good news is that my very good collaborator and fantastic experimentalist, Professor Valeria Clayman of Florida, it also happens to be my wife, or more precisely, I'm her husband. So that was a good weekend, right, when this happened. This guy is a different beast. If you see, there's meta-conjugation and then there's ortho, funny one. And what I like about this is that the organic chemists that made it, they really, truly, absolutely believe this thing to be flat. And they made all these ideas about this thing to be flat. I'm showing you here this. This is just a molecular dynamics simulation, nothing stupid, right? And this thing just is completely disordered. There's no way this is ever flat at any temperature that is interesting. What happened is the following in this particular case. That's the absorption, and this is absorption of the backbone, and that's the absorption of the perylene, right? And this thing actually fluoresces around 550. And what they've done is very detailed experiments where you excite at different wavelengths, where you actually take out the perylene. And experimentally, what they decided is that you needed at least the following rate constants. You absorb energy. There was this this intermediate that's created very quickly that then decays into excited state and then fluoresces. So the existence of that intermediate was predicted by experiment. And more interestingly, the prediction through transient absorption is that particular state was extremely localized in space, right? That the initial and the final states were kind of delocalized, the middle one was localized, and they didn't understand why. And if you look at the population of that state, it actually goes up and down very quickly. You can measure all the rate constants. Again, that's experiment. We did our job. We did essentially the same thing: excite, measure, look at all the dynamics, and what we get is this greenish one is the final state. This purple one happens to be the S2 state, right? And if you look at it, it goes up and down, right? The time scales are not quite correct, and that's just a question. I think we know how to solve that with some tricks in the dynamics. But the most important thing that we find is that the initial states are all delocalized in the 2 branches, and there is this S2 state where you always localize in one branch or the other, right? And then you transfer down. So you almost never ever see a transfer from delocalized to the end. You have to go to this gatekeeping state, which is the one that we think they found experimentally, right? Or we interpret it that way. So it turns out that in our case, it seems like a third option, or whatever you want to call it, where it's a delocalized exciton formation. Then you go through a gatekeeper state that is localized, that is mandatory, and then you transfer. All right. So how much time am I? Where am I? What time is it? ¿Qué hora es? ¿Qué hora? Oh. All right, I can slow down. All right, cool. So, all right. So the next thing I want to tell you about is completely different, except that it has to do with dynamics. Right? So I'm going to tell you about pH and pKa's, and it turns out that there is a cool way that I was basically talking to Jan Barber and Matt Jacobson at UCSF, and they completely blew my mind with this paper and some ideas that they have, where I used to think about pH as just something that happens, right? Cells have pH, reactions have pHs, tells you something about protonations, who cares, right? That's okay. It turns out that you can completely rethink it as a modulation, as a signaling mechanism, as a way that nature has to change behavior by very small changes in pH. So cells, normal cells have, some pHs outside and inside that are very close. When you actually get a metastatic cancer cell, the pH outside and inside are off by 1.5 pH units, right? And that completely messes it up. Nature doesn't want to do that. But more importantly, you can think about that you can have a protein that is perfectly happy doing whatever it wants to do in some part of your body that has pH 7, and then at some point this thing finds itself in a pH 6. And now it can start working, while when it's at 7 is inactive. This is a hell of a control mechanism, and it's very, very accurate, very precise. And what is interesting is that nature has a lot of these tricks, but if you think about phosphorylation, for instance, it's slow, you need an enzyme, you need to catalyze it, right? It's not naturally reversible or irreversible. Why? Protonation is just incredibly fast, milliseconds, right? Completely reversible, and it's uncatalyzed. So nature can play with buffers and change the function very effectively. And these things happen, if you're interested, come and talk to me about examples. Some of these things happen with changes of pH of about 0.4 pH units, right? Completely changes the way these things work. The way we teach general chemistry, it's pH, pKa's, is actually very different from what, let's say, biologists do. In PChem, or even in PChem, but in general chemistry for sure, we have an acid or a base in a beaker. We have an electrode, right, that measures protons. That thing is designed to measure protons. And then what you do is you add acid or base, you look at the needle, right, and you see when it changes. And then you go, boom, that's a pKa. Right. Let me show you a biological experiment. That's heat shock protein 90. It has two different states at two different pHs. At low pH, this thing is closed. At high pH, this thing is completely open. So you can do an experiment where you measure size, essentially. That just, it turns out that it's a Sachs experiment, but you measure something that has to do with size, with shape, versus pH. You get a sigmoidal curve, great, right? And you get a halfway, and you call that a pKa. This is a perfectly good definition, thermodynamic definition of pKa. pKa is the pH at which half of that happened, right? And we obviously believe that there is a proton involved somewhere, otherwise this thing would be flat, right? But the measurement has absolutely nothing to do with protons. There's no electrode here. It's some other measurement that reports on conformation. And that's important because you can get yourself in big trouble by doing this. Who can be responsible, right? There's a small number of things in proteins. Amino acids, nucleic acids have their own particular set, but there's really a small number of, arginine, histidine, lysine, aspartic glutamate. You can add tyrosine and cysteine to this thing. They don't have their own pKa's in solution, right? These are the building blocks. There's not that many things that nature really uses a small number of building blocks. Most interesting things happen about pH 6.5. That's where we live, right? 7. So if you're going to have something interesting happening, it has to basically go around that number, right? So what people do, and I have another thing is, for those really interested, people really go and basically try to find a histidine because they say, that's the one with the proper pKa, I'm going to find it, right? And I'm telling you now, we can discuss this later, we're finding paper after paper after paper where that is absolutely wrong, right? It's not the histidine. The amount of energy you need to get an aspartate to pKa 6.5, it's 3 kcals per mole. That's nothing. That's chump change in biology. So what we want to do is we want to control pH in simulations. Right? We want to do molecular dynamics where pH is a variable. And the way I think about this is it's a long history. None of it is mine. This is 40 years of research where we did not know how to do constant temperature. So we invented the thermostat. We did not know how to do pressure. We invented the barostat, right? And in some sense, what you're doing is just bringing the techniques closer to experiment. Instead of putting the protons where you think they should be, you said, look, I'm going to do like an experiment. I'm going to put a buffer. And the proton can do whatever it wants to do. What can you do if you can do this, right? You can look at pKa's, changes in conformation, folding, a lot of things that are immediately interesting. So it turns out that if you look at pH modeling, we've been lying to people for like 30 years, okay? So you see papers that say, experimentally it's low pH, high pH, we're going to run simulations at low pH, high pH. And what they've done is they guess. At low pH, these things are protonated. I'm going to put a proton. At high pH, they're not, right? Sometimes the guess is good. Significant number of times, our experience is reasonably good. But let me give you an idea. If you have an aspartic acid, pKa 4-ish, right? And you happen to have an experiment around pH 4, I guarantee you're wrong 50% of the time. Yes, exactly. So that's bad. But the other one is that it turns out the pKa sometimes is not a precisely well-defined thermodynamic object. It's very subtle. But more importantly, if you have an aspartic acid, you think it's 4, and the damn thing is 6, right? Now, if the experiment is done at 6, then you're screwed. You don't know what to do, right? Any choice you make is going to be wrong. So it's a bit of a problem. What one should do, and I'm waiting for Alan to do it so I don't have to worry about these things, you basically need to simulate the following equilibrium. You have an acid with a proton in water. That proton goes away, proton migrates away, and you need to hydrate it properly. Each one of those 3 problems is insanely hard. Right? So what we're going to do is we're going to forget about it. We're going to do a complete trick. We're going to not do that. Right? So the way this is solved is the following, and I'm going to do this in 2 steps. We're going to do a mixed Monte Carlo, molecular dynamics Monte Carlo, and we can show all that the stat mech of this is correct. I'm going to start a molecular dynamics simulation with some residue. It's hard to see, and that aspartic acid happens to be protonated. I just pick one, doesn't matter. I'm going to run molecular dynamics with the proton there, right? So everybody moves. And then I'm going to ask the following Monte Carlo question. Can I take that proton away? Yes? And that question must come essentially from 2 factors. One is the pH. So if the pH is, 2, chances are that the answer to this is going to be no, that the proton is going to stay there. But also it's going to depend on the local energetics. Yes? So those are the 2 factors that will give you that answer. Again, Monte Carlo that's a great news, 2 answers, yes or no. So if the answer is yes, I now keep the proton away, run molecular dynamics without the proton, and at the end of this, I ask again. But now I ask, can I put it back? Right? And you keep doing this. If the answer was no, I run more molecular dynamics. Sorry, I need to go forward. Protonated for some time, and then I ask again, can I take the proton or not? When I'm done with this, I'm going to show you how do I answer that Monte Carlo question. When I'm finished with this, what I have is a set of structures. Some of them have protons, some of them do not have protons. I know the pH, so I can do a titration curve. If I run this simulation at a different pH, I get myself a beautiful titration curve. I can ask whatever I want. Right? How does this work? So it turns out that where what I really need is the free energy to take the proton away of a residue inside the protein, right, for instance. And to do that, I need to break the bond, let the proton go away, hydrate the proton. I told you I don't want to deal with that. So what we do is a nice thermodynamic cycle. With what I do is, essentially, I take away the protein. I get the amino acids alone in solution. I take the proton away, and then I put the protein back up, right? Like, Hess told us that if I can do those 3 legs and I add them up properly, I get the same answer. That's the good news. How do I do that? The top is actually very simple, the top left and the top right. All I need to do is compute the interaction of my system with that residue protonated and unprotonated, right? And we know how to do that. That's the core business of force fields. The bottom one happens to be essentially related to the free energy of getting an aspartic acid, protonated and deprotonated, alone in solution. But that number I know experimentally, right? I can fit to it. I can figure out that that's my reference. And then everything I do is basically relative to that number. So what I'm doing is computing delta pKas, not pKas in some sense. The great news about this is that some of the things I told you are approximations, but the free energy of the proton in solution actually goes away analytically. I don't have to worry about it ever again. This is non-trivial. The free energy of solvation of a proton has error bars of about 3 to 4 kilocalories per mole. That's essentially half the pH range, right? If that's the error bar in that number, you're screwed. You can never actually get an absolute pKa. All right, so just to give you an idea what it is, this is a model system, a fake system, right? This is 5 glutamic acids in a row, and what we do with the number is very, very simple. I'm going to say, do this at pH 4, right? And at pH 4, what you see is things moving, and the protons, sometimes they're there, sometimes they're not. Okay? So now, if I do this thing at pH 7, the only thing I change in the whole simulation is just that number, 4 to 7. And when you look at it, you basically see never a proton there, right? Once in a while there's one. And that's because the pK of these things happen to be around 4.5 or so. Right? So at 7, this thing should be deprotonated. So this is the kind of things we're looking for, right? This is what I mean by constant pH molecular dynamics. The ones that have been paying attention will notice that I do dynamics on Monte Carlo. So even though this thing looks like a movie that is continuous in time, it's not. The Monte Carlo step just takes away that, right? So one should not interpret this thing as dynamics. It's an ensemble generator. So let me show you a little bit. This is a very nice paper. Bertrand Garcia Moreno at Hopkins has been making a business about the following. He took this staph nuclease protein, mutated about half of it to make it tremendously ultra stable. This thing is a rock, right? And it has a very large number of hydrophobic residues inside. And then what he did is he messed with it. He basically says, let me go in there, and where I used to find the hydrophobic residue, let me put a charged residue. Let me put a lysine, let me put an aspartic acid. And this thing is not going to like it, right? So if you measure now the free energy of unfolding of, the wild type and then the mutant with this charge, you can get an idea of the pKa of this residue that you put in there. These are some pKa's measured by them by lysines, right? That he put in there. And are those the lysines? Yes. And if you look at it, the pKa of a lysine alone should be 10.4 up there. Some of the mutants have pKa's as low as 5.2, 5.3, right? Completely away from they should be because they happen to be inside. The idea that Bertrand had for years is the following. I'm going to have a hydrophobic environment. Water cannot get in. This lysine will not like to be charged. Yes? Which means the pKa will be very low. If you can do this calculation in 20 different ways and you get always the same answer. The pKa of that lysine inside the hydrophobic pocket is -5. Right? It's incredibly low. This thing really, really, really does not want to have a proton. Right? So even 5 looks impressive. The story cannot be that it's just, that it's in a hydrophobic pocket and doesn't want to have a proton. Because if that was true, that pKa will be much lower than even 5. And then you get all this, this selection, so it's a nice setup. So what we did is, we did our method. We actually do for the experts. We can do pH, molecular dynamics with replicate change on top, so we can really make sampling much better. Those are a whole bunch of mutants, and you get titration curves by doing this at different pHs. One thing that I kind of proud within my group is the error bars in the computational part. We basically, at this point, we run 5 to 10 of these simulations independently. Doesn't matter what we do. We just run 5 to 10, and we average and we do statistics, right? So those are mutations to lysine. The ones on the right are mutations to glutamic acids. If you do the calculated versus the experimentalist, that's the correlation we're getting, right? And this is just, I don't trust the data that much, okay? This is just too good. Just to show you that I don't trust it that much, there is an offset of about 1 unit, so the correlation is great, but we're off by 1 unit consistently, and I don't know where it comes from. I'm looking for it, but you know, this is not the end of the story. It's really good, but it's not the end of the story. We can do these things now. 1 mutant is about half a day. 5 replicates of this. This is really, really quick. Oh, it's all in GPUs. Right. So now we're interested in something, those two guys. Those two guys are very interesting because they essentially have roughly the same pKa experimentally, and they're in completely different places in the protein. And there's evidence that the reason why they do this is really different in one case and another. So we can titrate that lysine. We get ourselves a titration curve. We get ourselves, you know, the experimental was calculated. Again, that's just too good. All right? I'm just showing off. There's no way that the method is accurate. It's not precise, I'm sorry. So, and then you look at the data, and there's something very interesting. This lysine, we run it at different pHs, and you find the following really nice thing. At pH 12, this thing is completely deprotonated. We know that. All right? We can look at that. And we look at the dynamics, and this thing is completely buried inside the protein. That's okay? Because it's deprotonated, that's where it doesn't want to be in water. But I told you before that when this thing is deprotonated and it's inside, the pKa should be very low. So we have this high pH, and the pKa should be very low. They're opposite. Okay? You can look at, you know, the environment around. There's nothing around. I mean, there's stuff, but there's no water around it. The number of waters versus time is very low. This thing is dry. It doesn't want to do anything. Right? Now you look at the same thing but pH 3, the other end. And I can tell you that this thing is protonated. We can look, right? It's completely solvent exposed. Now it's looking outside. It's sticking its head out. And if you look at the number of waters, look at, you know, the black. That's the first solvation shell. You get continuously 5 to 6 waters in there. So now the pH is very low, 3. The pKa is, you know, high because this thing is protonated. And before, the pH was high and the pKa was low. What's interesting about this is that if you think about it, whatever is it that you're seeing is not simply a protonation-deprotonation equilibrium, but there is a conformational change associated with it. When you change pH, you change protonation, you change structure. And so that means that to think of the pKa is a well-defined thing. It's when half of the things happen. But to think of the pKa as the free energy of protonating, deprotonating, is just simply wrong in these cases. The free energy you're looking at is protonating, deprotonating, and changing the conformation, right? And you need to take that into account. So there's one more thing. What I've done is, for that system, we measured at very low pH, solvent accessibility of that residue. Green curve is high. This thing is solvent exposed, doing its own thing. At very high pH, this thing is very low surface accessible area. If you actually measure, you histogram this thing and you integrate, you say how often this thing is inside, how often it's outside, and you look at that thing versus pH, you get a sigmoidal curve. That matches the one that you did for protonation, right? These things are coupled equilibrium. So it turns out that in this simple system, you can go back to basic pChem, and you have actually, you need 4 states. You don't need just 2, protonated, deprotonated. You need, let's say, a closed state, protonated with its own pKa, an open state, solvent accessible, with its own pKa, and then you have an equilibrium constant of the free energy that is associated with a conformational change, right? And you can actually solve this essentially for different values of the equilibrium constant. You can also measure it and you can calculate it. And you get yourself this pKa observable, this macroscopic pKa that is some weighted combination of the two microscopic pKas. Let me spend 2 minutes on the following. It turns out that, and Alan will look like this. If we can do constant pH, we can do constant electrochemistry. Instead of exchanging a proton, we're going to exchange an electron. Who cares, right? The code turns out to be essentially identical. And, instead of a log, you get an ln. So instead of Henderson-Hasselbalch, you get the Nernst equation. They're mappable to each other, right? There's just constants in front, right? So what we can do now is do titrations, electrochemical titrations. We can look at electrochemistry in these things. Just to give you an idea, this is just an example. We did this about 2 weeks ago. The curve in the middle is fraction reduced versus redox potential. So if you go half to this, you get E naught. The black curve is iron 2, iron 3 in solution. All right? So you get yourself 0.77 is actually the E naught measured, right? And then we get the same number. If you now put charges, positive or negative, sodium plus 2, sodium minus 2, good thing with computational chemists, right? We can affect these things. You actually see these shifts, right, to higher or lower electrochemical potential because the environment has changed. This is just the first step we needed to figure out if we can do this inside the protein, inside some other things. If we do this, we can look at mutations versus, E naught versus mutations. We can look at protein-protein binding, ligand binding. How does electrochemistry and pH relate to each other? This is called the redox-Bohr effect, right? And what is interesting is that I don't know exactly how to, but I think we can split. We can do water splitting because we can look at these proton-coupled electron transfer processes where you actually have states. The one at the bottom is the simple one. You get a very nice equilibrium state where the proton happens to be. Let me look at this. The proton happens to be on the right side, right? And it's going to transfer. And if you just transfer a proton one side or an electron the other way, they're unstable. But if you transfer a proton one way and the electron the other way, the system becomes stable in a new conformation. And again, technologically wise, we have this constant pH, constant electrochemistry things, but there's nothing against doing it at the same time, right? And we're basically ready to go out and do those kind of things. Let me show you, that's my group. This is about Alan's group size by now, so that's a good thing. So that's people that put money. Collaborators in Argentina for many years, Dario and Marcelo, and I collaborate in the non-adiabatic things with Sergey and Sebastian. And the last thing that I want to say, just as a final joke, is when they originally invited me, they said, my talk was going to be right before Calculus. And I don't know if I talked to you this. I was going to be el telonero de Calculus, right? I was going to speak right before him. So now Alan spoke right before me, so there's some symmetry to that. Thank you, and questions.
Okay, when you have this this protonation or this question if it is protonated or not.
Yeah.
So my question is, when you have a carboxylate, so how do you decide with oxygen you are going to protonate? And the question is that relevant? Perhaps it is not.
It's a great question. We say, carboxylate has 2 protons, so it's deprotonated. And now he asks, can I put a proton? Where do I put it? Right? Yeah. And that's simply a question of, let me go back. I can put it anywhere I want, because eventually if I put it on this side, the dynamics will do this, right? And it's fine, right? So that, in a formal sense, it doesn't matter. In a Monte Carlo sense, to make it more efficient, we actually go from deprotonated to 4 possible protonation sites. So we make a decision to go like this, like this, like this, or like this, right? So we pick one of those moves. But that simply makes it more efficient. It doesn't really change anything in the thermodynamics.
Okay. Can I just... And when you said, okay, so I can do pH and then, acid-base equilibrium, and I can also do electrochemistry. So when you have this potential, so you can also change, for example, the oxidation state or of a species depending on the potential you're working with.
Correct.
Okay.
Correct. And again, the goal application, again, I come a little bit from biology, is there are these proteins that when they're separated, their redox potentials are such that you will have no electron transfer, but when they bury the interface and the water goes away, the electrochemical potentials shift. So now you get efficient electron transfer. Those are the kind of things we want to look at. Changes on electrochemical, on standard electrochemical potentials when you change something.
Thank you.
I have one technical question.
Sure.
And it's, how often do you protonate or deprotonate? Because I know we do this for binding, protein binding, that we don't actually look at the molecular binding, but we just make them bind or unbind. And the thermodynamics doesn't matter on the barriers, but you could actually do dynamics with this.
You can do what, sorry?
You could do dynamics with this. And there the timescales could actually be interesting. Or in reverse way, I don't know if you ever thought about this, could you learn from this how fast this protonates or deprotonates by changing your rate?
Those are great questions. So the first one is, yes, of course it can be done. If I try to protonate, deprotonate somehow in the experimental timescale, then I'm essentially looking at dynamics. The problem is that that timescale is essentially milliseconds, so we're dead. Right? I'm not going to wait dynamics milliseconds to change it.
So the barrier
is the barrier, and it's the fact that you actually you transfer a proton to water, and if it happens, it's a very funky water mechanism. Sometimes it comes back, so that kind of doesn't count. And the other time, if this thing goes Grotus, it goes boom, and it shows up over there very quickly. And it's that balance, right? Deuterium exchange experiments tell you a little bit about the time scales of these things, and they're systematically millisecond-ish. So they're too long for us. Yeah.
Now my question is, like Tomas was asking, then what if you have a pocket with many residues that can be protonated or deprotonated?
So if I have more than one residue, right? So the question is, what if I have more than one residue that can protonate, deprotonate? Let me give you two extremes. One of them is they're very separate in space, then it doesn't matter, right? They're independent. We studied a lot. The active site of HIV, classic example, it has two aspartic acids, right? Very, very close to each other. And the question is, how come, right? There's no way. And what happens is they actually share one proton. One of the two guys is protonated, and that's enough to make this state. And we tested that in many ways. So what happens is, if I take, let's say they're both deprotonated in my simulation. If I try to change the protonation on this guy alone, right, it's not going to work. If I try to do this guy alone, I'm sorry, this is protonated, this is not. If I try to take that proton away, it's unstable. If I try to put a proton on this one, it's unstable. So I need to change them both at the same time. So we have a Monte Carlo move that looks at residues that are coupled, and once in a while tries to change them at the same time. Yes? In principle, there's nothing, if there's 3 or 4, that makes it more complicated, but just faith in Monte Carlo.
I have a question about the actual E zeros of the amino acids, and eventually you're going to put the protein in a certain peak, a certain E zero, a certain potential, and then you're going to drive the reduction oxidation of the amino acids themselves.
Correct. Well, right now we're looking at cofactors. It's a little bit easier right now.
No, but I'm asking you about the amino acids. The cofactors, for example, as we have these databases, we know more or less how much is there,
that's why
errors, etc. But then how much is known about the amino acids? Can you, even as you say completely correctly, pKa changes so much in different regions of the protein?
That's right.
Maybe some amino acids are doing redox and we don't even know.
There's a small number of amino acids that can do redox. There's basically, so cysteine, absolutely, right? Of course, yeah. Tyrosine can do quinones and things like that. The other ones, you can get a free radical for tryptophan under very special circumstances, and eventually we'll go there. We don't know. Let me give you the answer. As it turns out, in some sense it doesn't matter. If I choose my reference compound correctly, right? Let's say a tryptophan in solution, and either somebody measured the E0, or I look to your database, or I have no idea what it is. right? If I now compute delta, what I can say is, I don't know the E naught, but I can tell you that it goes up by 30 millivolts if this happens. And for now, I'm okay with that answer.
Have you detected any problems using hydrogen micropartition?
Well, no, that's too technical. We'll talk about it later. He's asking me about funny. We published a paper last year where we actually showed that in molecular dynamics you can double your time step. You can go from 2 femtoseconds to 4 femtoseconds with exactly 0 effects, right? It's a little mathematical trick, and we put this out as a paper asking people, please break it, because I didn't believe it. And we'll talk about it, but reports from the ground says that it's working, and I don't understand exactly how.
More questions? Agradecemos al Chicos, esperen un momento, por favor. Sí. No sé si... No. Well, our symposium has come to an end. Our symposium celebrating the 100 years of our School of Chemistry and the 75 years of the Institute of Chemistry. Well, in these 2 days we reviewed theoretical aspects in density functional theory. We had also talks about the nature of the chemical bonding, quantum chemical topology, nanocompounds. We also had applications in biological systems, etc., molecular dynamics, excited states. I think we had a very rich variety of talks. We are particularly happy of our poster session. We had 45 posters, students interacting with professors, professors with professors. We really liked it. The students, especially the youngest ones, had an opportunity to interact with experts in the area. Yeah? While in Spanish, a los estudiantes que por primera vez asisten a este tipo de eventos, sobre todo los más jóvenes, espero que les surja la inquietud y se acerquen a profesionales de la química teórica y exploren si les gusta la teórica o la computacional, ya sea aquí en la facultad, en otros lugares de la UNAM o fuera de la UNAM o incluso de México. Dense una oportunidad y vean si por ahí van sus intereses académicos. To our visitors, thanks for coming and share your knowledge. Thanks for coming to Mexico. We are very happy of having you here. I hope you have a good memory of being here. And to our colleagues and students from Mexico and outside, but also in Mexico, thanks for your interest and for taking part in this symposium. So, thank you. Bye-bye.